viable option to solving the protein folding problem. And I know many of us in the room may not know much about machine learning. And honestly, to be fair, 4 years ago, I didn't know how to use my laptop. But, you know, as a double major, I've been able to better understand how technology can impact the natural sciences. And kind of the inspiration behind this is just over the past 4 years, I've noticed the techniques and methods that we were doing that could be done more efficiently. And through my presentation, I hope that I can convey to you the importance of machine learning. So before I go into the more fancy stuff about machine learning, I just like to talk about the current states of determining protein structure. So the current methods used are crystallography, monochrome electron microscopy, NMR, molecular dynamic simulations, and in silico prediction methods. These methods are techniques that even I was able to use as an undergraduate, so I really won't be going into too much detail about them since many of us are familiar with how they work, but it's important to note that molecular dynamic simulations and in silico prediction methods are both methods that are a combination between machine learning and benchtop techniques. So with these, they have their own strengths and limitations. So their strengths is that there's no data dependency. In essence, you're kind of creating your own data. And you know, certain tasks are irreplaceable, especially when it comes to the X-ray and microscopy techniques. You know, you can't image with a computer, and imaging these samples holds a large amounts of information. So with these strengths, it's quite obvious that, you know, the well-worn way of discovering proteins is irreplaceable. These are things that a computer can never replace, but they have some pretty big weaknesses. They're time-consuming and expensive. So I don't know about you, but I do not have $7 million offhand to buy a cryo electron microscope, and nor do I have the space for it. So you're really dependent on that hardware and space and for an institution to provide those for you. And I think this dependency on institutions was really evident. That's like kind of a weakness in COVID, as you know, labs have been shut down and research had to come to a halt during the pandemic. And even here at Chapman, you're also very dependent on samples and materials. So you're dependent on how much sample you have, the materials you have at your disposal, whether or not you can store those materials, all different things that come into play. And then there's also the knowledge barrier. I just like to note that, like, as a 4th year biochemistry student, I have zero confidence in the fact that I could go from sequence to structure all by myself. I would definitely need, you know, the help of someone like Dr. Owens to help me get from point A to the end, which is not the same for machine learning. So now that I've kind of addressed the strengths and limitations of the current methods, I want to move more into the machine learning aspects and explain some of the specific models that are most commonly used in the protein folding application. But before I do that, I'll just simply define machine learning as, you know, finding patterns out of data, making sense of those patterns, and then using them to your benefit. That's what machine learning is at its very core. So first we'll get into support vector machines, or SVMs. So how SVMs work is they are a binary classifier, which means they classify into either or, and they classify using this hyperplane, which makes their data linearly separable. So in terms of protein folding, if class A was alpha helices and class B was beta sheets, it's actually able to plot based off of the pattern of amino acids. So as we know, alpha helices, the amino acids are arranged in a different pattern as the beta sheet or beta sheets. So it's able to classify those two different structures on this like plane and be able to make them linearly separable with this hyperplane. So next I'll go into neural networks. So neural networks is definitely more of like a buzzword. This is more of the famous example. And how they work is, if we wanted, I'll explain it with a very simple example. So if we wanted to train a data set to be able to identify cats, dogs, and fish, we'll input all that data into this input layer, and through a series of computations in the hidden layers, we'll get an output that actually gives us a series of probabilities of how likely it is to be one of the different targets. So say we train this with those 3 animals and we put in a picture of a dog. As it moves through the hidden layers, it'll probably give us an output that's something like it's 0.7 probability of being a dog, 0.2 probability of being a cat, and 0.1 probability being a fish. And we're like, yes, I put in a picture of a dog, therefore that's correct. The answer should be a dog, right? But I think neural networks display the idea of learning best because actually when this initial movement starts, the parameters are actually randomized. So it actually hasn't learned yet the picture. So it'll probably say, like, if you put in a picture of a dog, it'll probably tell you it's a fish. But as we all know, a fish is not a dog. So we can tell the computer that's not right. And it'll go back and forth and adjust the parameters that make the computations to eventually give us the right answer that tells us, yes, a picture is a dog. The most notable example of neural networks in protein folding is AlphaFold, but I will get into that later. The next example of machine learning I'd like to show is the hidden Markov models. So hidden Markov models are more of a specific application of determining protein structure. So SVMs and neural networks are more generalizable, whereas hidden Markov models excel when being applied to DNA binding proteins. They're incredibly powerful and very commonly used in the world of genomics. They're actually able to create predictions based off of what came before, which is important in terms of genomics because then we can then predict nucleotides based on what came before, right? And this example was really just to demonstrate that just like the current methods, there might be a more like niche model that's going to be able to achieve better results and than the more generalizable ones. And hidden Markov models are actually really commonly used in tandem with SVMs to achieve more accurate results. So now I'm going to just talk about some of the strengths of machine learning. The cost of services is relatively low. And when I mean relatively low, relative to $7 million for one microscope, you know, a good graphics card can cost you about $2,000. And that's the computer component that does a lot of the computations. And with this comes with the hardware benefits. It is relatively easy to store a pretty powerful computer. You don't need too much space. But if you didn't even want to worry about the hardware, there are cloud services that you can host all of your data and information on, like IBM Cloud or Microsoft Azure. And with that comes with accessible support 24/7. And another thing that's really unique kind of to the machine learning and the computer science world is open source. And open source is when proprietary knowledge is available or free to access. And this has kind of created a community of working together, finding new solutions and sharing information. And this kind of has led to the accessibility of education as well. So platforms such as Coursera have been able to provide low-cost education that's accessible to the public. And then the specific strength that I want to just showcase that's an example of how powerful machine learning is AlphaFold. So AlphaFold is the protein prediction software that was developed by DeepMind, which is the research sector of Google. And they created this prediction software, and it competed in the critical assessment of protein structure prediction competition, which happens every 2 years. And CAS basically gives you a sequence and it's like, find its structure and we'll tell you how right you are or not. And it's an international competition. And the graph to the left shows the median free modeling accuracy, which is the overall accuracy of your structure. And from 2006 to 2016, we can see really no general trend of things improving. It kind of seems more like sigmoidal in like the trends, I don't know, but there's really no general overt trend of improvement. But when AlphaFold first competed in 2018, it actually already showed to have a 20% higher accuracy than the year before. And then when competing again for the second time in 2020, it achieved even 30% increase in accuracy. I think this goes to show just like how powerful computers are, that within 2 years, it's able to achieve a greater improvement to accuracy than ever before. I mean, no other year compares. And to the right is actually a picture of their 2020 results. So in green is the experimental result that CAS dictated was the right answer. And the blue is the computational prediction that AlphaFold had developed. And of course, with strengths comes limitations. There are limitations within the models themselves. I mean, SVMs don't do well with large data sets or data that isn't linearly separable. And neural networks and SVMs are both prone to overfitting, which means that your model is too specific to your data. So when it comes to your data and running your data and your testing data, it's incredibly accurate. But the moment you integrate something else, it's not so much. You're very dependent on data. As we see to the left, this is like an example of choosing incorrect data and like just the biases that come with it. There are also knowledge barriers, but I did address the fact that, you know, Coursera is available and there are ways to get educated without, you know, going back to school. And there's also a lot of hesitation within the community, but to be fair, with things that we've used like CCP4 and Primal, they're really hard to use. And I don't think with reason, but with that being the current state, I understand the hesitation. So now I tried to talk about some translational applications of machine learning to protein discovery. So first we have drug discovery. This figure is just a bunch of machine learning models and their applications in drug discovery. But some notable examples of machine learning in drug discovery is molecule design, molecular docking, biochemical interactions, genomics, drug sensitivity. And as opposed to having scientists come up with all possible structures for a specific drug, machine learning models are being leveraged to propose the most likely structures for these drugs. This in turn is making drug discovery process more efficient. And the next example of translational applications is precision medicine. So this figure is also demonstrating how machine learning is being integrated to improve diagnosis, prognosis, and therapy guidance for patients. And some notable examples of machine learning in precision medicine are predictions based on electronic medical records or EMRs, targeted personalized treatments, cancer classification by tumor cells, and genomics. Being able to make medicine more personalized is a step in the right direction in terms of patient care, as medicine kind of in this industry is a one size fits all type of situation. So as I conclude, I just kind of want to leave you with one question. So machine learning has become such an integral part of our daily lives. I mean, we use it into like self-driving cars, social media, speech to text. In really every aspect of our lives, we use it. So why is there so much distrust of it in the scientific community? And now I just like to thank Dean Eva for the never-ending support throughout my capstone. It was so much fun, and I hope you also had fun. And then to thank Dr. Eric Lindstedt and Sonica for supporting me over the past 4 years in computer science. And I'm open to questions. Thank you.
Amazing. Let's give Kaitlyn a resounding round of applause. Definitely, if you use your, feel free to use the emoji as well, but that's also a nice little added touch in Zoom. Kaitlyn, wonderful, wonderful talk. I believe we have a couple of minutes for questions, so maybe 2 or so questions we can entertain right now. So please go for it.
Can I ask a question?
Please.
Kaitlyn, fantastic job. Really appreciated this. You asked the question on your last slide. Do you have an answer?
Do I have an answer? I mean, personally, I don't understand the hesitation. In my mind, I dove like kind of blindly and headfirst into computer science as well. When I came to Chapman, I was a biochemistry major. I didn't even know how to use my computer. Like, I'm being serious. And like, I think, you know, like, to me, like science is about like learning things you don't understand and kind of going into that head first. Like, I mean, isn't that the whole point of science, is to discover new things? So like, why not adopt new things instead of just like relying on like what, if it still works, like don't fix it, you know, if it ain't broke, don't fix it. So I don't really know why it's a hesitation. I mean, personally, obviously, I love computers and I love machine learning, and I think it's really interesting. But I don't know. I'm not the hesitant one, so I can't really give you that answer.
Thank you.
We got time for one more question. Yeah, I think, Kaitlyn, maybe what I would say is that, as you mentioned, science is all about learning new things. Scientists, on the other hand, are a different story, and that is certainly worth further investigation. Well, let's go ahead and thank Kaitlyn for a wonderful talk again. And congratulations, Kaitlyn, on a fantastic undergraduate career. And we're looking forward to having you back again for a master's in your computational and data sciences. Okay, wonderful. And our next talk is by Charis Thompson. Karyss is has graduated, of course, with a bachelor's in biochemistry and molecular biology with a minor in political science, and that is an interesting combination. She worked with Dr. Mike Eba as well, also trying to study how to cure mental health disorders one microbiome at a time. That sounds fascinating. Karyss, take it away.
Thank you. So like Dr. Ogba said, my capstone is titled Curing Mental Health Disorders One Microbial at a Time. So for my capstone research, I did a literature review, and the goal of the review was to explore how the microbiome contributes to the manifestation of mental health conditions, and then to determine if there was a potential for new treatment given this connection. So the mental health conditions that I primarily focused on were anxiety and depression. I have a few other different conditions, but those are the ones that I mostly address, as those are the most researched in this field. So the next couple of sections, what to expect out of this presentation, I am going to give a kind of background information and explain kind of the evolution of our understanding of the microbiome, and as well as mental health disorders, the research that I looked at to discover the connection between the two, and then finally how this connection can be used to discover treatment options. So the microbiome is the microbial cells found in all humans. There's between 10 and 100 trillion microbial cells in the human body, which is roughly 10 times the amount of human cells in the human body. And so the these microbes live in a symbiotic relationship with their host, helping to just make normal functions of the body work, like digestion, just regulatory functions. So the first mention of the microbiome was in the 1880s by pediatrician Theodore Eschrich. And he actually found E. coli was present in both the intestines of healthy children as well as those suffering with diarrheal disease. And so from there, scientists began isolating microorganisms from all over the body. 2001 was a really big year for the microbiome. This was the first formal definition of the microbiome by Lederberg and McCrae, and they described it as the ecological community of commensal, symbiotic, and pathogenic microorganisms that literally share our body space. In the same year, Wellman and Falco sought out to create a microbial inventory of all the different areas of the body, and they found that the genomes of that make up the human body consisted of bacteria, fungus, viruses, but mainly were bacteria. So then the composition of the microbiome is different in all different parts of the body. The gut is where most of the bacteria can be found, and is where most of my focus on the microbiome is. So the factors that influence the composition, it starts with birth, and so even the mode of delivery is really important with building the composition of the microbiome. So individuals born via natural births versus cesarean births, you're going to have more beneficial bacteria if you were born in a natural birth versus a cesarean birth. And then it continues on with even how you were fed as an infant. In general, individuals who are breastfed versus formula fed are going to have higher beneficial bacteria in their microbiome. Then things like diet and environment begin to play a huge factor in continuing to build the composition of your microbiome for well, even now as an adult, it plays a factor in your microbiome. So with diet, it's the things you eat feed the microbes in your gut as well. And so if you're not eating foods that have the nutrients that the microorganisms need, then the microorganisms are not being fed, and then they will eventually die out. And then with the environment, microbes are not trapped inside our body or inside the air. They flow freely between humans and the environment. And so everything you touch and come in contact with has the ability to influence the composition of your microbiome. So the microbiome has been found to be associated with many health conditions, including obesity, diabetes, cancer, cardiovascular issues, liver, renal diseases, have all been found to be associated with microbiomes that are not functioning, disruptions to microbiomes. And they have found that treating the microbiome issue helps to decrease the severity of these health conditions. And so this same kind of approach has been taken on with mental health conditions now. So mental health, I'm sure you guys have heard this term before, no health without mental health. Mental health is incredibly important to all aspects of our life. But there are three theories that throughout the years have been recycled in an attempt to explain where mental health conditions come from. And so the first real explanation was this supernatural theory. And so the first treatment methods that they used for the supernatural theory was skull drilling. And then if you've ever talked to anybody that's in the Catholic or Christian religion, we do things like confession. They believe with the belief that mental health conditions manifest from an idea of like an all-powerful being from God, from a supernatural entity. So things like exorcisms have been used to treat mental health conditions from this belief and from this theory. The next theory is the psychogenic theory. And so that's the idea that it's all in the mind. All mental health conditions come from the mind, can be treated from the mind. So treatment options on this theory are hypnosis and therapy. And then there's this somatogenic theory, and that is the idea that it's a physical condition that causes the mental health or the manifestation of mental health conditions. And so the first example of this was hysteria in women, and doctors believed that the reason for hysteria in women was because their uterus was out of place. And so they had to smell this certain smell, and that would help to bring and guide the uterus back to place. So my theory that the microbiome contributes to mental health is more of a somatogenic theory. And nowadays we kind of practice somewhere in between with a psychogenic and somatogenic theories to treat mental health. So making the connections between the microbiome and mental health. So the connection between the two lies in the gut-brain axis. And so the ENS system, it is part of our intestinal tract. And so there are receptors in there that actually are for the neurochemicals that are produced by microorganisms. So then the ENS then communicates with the CNS, which then communicates to the brain. So this gut-brain axis provides the ability for bacteria to communicate with the brain. And so the first kind of indications of this was with inflammation and mental health. And they saw, so the first reports of this was in 1990, and they saw that the increase in severity of anxiety and depression was associated with an increase in inflammation as well. But they was not really sure which one caused the other. So they even did this study about taking NSAIDs in addition to antipsychotics for schizophrenia. And they saw that the use of NSAIDs in addition to antipsychotics showed a reduced a reduction in symptoms more than just antipsychotics alone did. So then you see the inflammation in the microbiome. And so there's been studies that show that there is an association between gut dysbiosis, which is a disruption to the microbiome or an imbalance in the microbiome of the gut bacteria. And that has led to an increase in inflammation as well. And they believe that this is due to a depletion of beneficial bacteria within the microbiome. And so this depletion is most likely due to 4 things that happen. And one is the overuse of broad spectrum antibiotics. Just in recent times, we use antibiotics a lot, and we use this like broad spectrum antibiotics, which don't target specific bacteria. They kind of target all bacteria with the same, or most bacteria with the same affinity. So it's not just targeting the bad bacteria, it targets all the bacteria, and so then that leads to a depletion of even the beneficial bacteria. That's why a lot of times doctors recommend taking a probiotic when you're taking an antibiotic. Then the other thing is poor diet, and poor diet for the microbiome is one that just doesn't give the microorganisms in your stomach or in your body the nutrients that they need to thrive and live. So this is something like a low fiber diet. So then the environment, similar to how it played in a role in the composition of the microbiome, same thing. You touch things, it changes your microbiome composition. So then my research is divided into 3 different parts, just mental health and the microbiome in general. And so I looked at 4 studies, 2 of which were using probiotic and prebiotic intervention. And they both saw that with the use of probiotics or prebiotics, that the severity of depression and anxiety symptoms were significantly reduced. And then the other 2 were fecal microbiome transplants, and similar results that actually like transplanting healthy microbiomes into individuals help to improve the severity of anxiety and depression symptoms. So then the next focused on mental health, the microbiome, and inflammatory bowel disease. And so it just showed the kind of just connected how inflammation and mental health and microbiome are connected, and led to the research on the just general inflammation. And so it said that, so it showed that the individuals with less diversity in their microbiomes or less beneficial bacteria in their microbiomes had more severe symptoms of IBS. And then finally, the big stuff, connecting mental health, microbiome, and inflammation. And so these studies found, the first 2 studies are just kind of general, found that mental health, that the microbiome of individuals with mental health conditions was different than those without, and that the inflammation was increased in those with mental health, with increased anxiety and depression symptoms. And then this last one, Mays et al., is a really big one. It looks at the idea of bacterial translocation and how bacterial translocation contributes to the cause of inflammation. So that is the big picture of this big idea, is that a gut dysbiosis causes increased gut permeability. One example of that is less short chain fatty acids is going to increase the gut permeability. And so that could be due to a low fiber diet, and which causes a change in the microbiome and to less bacteria in your gut producing those short chain fatty acids. And so then the bacteria in your gut moves out of it into places in the body where it's not supposed to be, which causes an immune response, because essentially it's a bacteria infection. And that's where the inflammation comes from, is from this immune response. And the immune response is what causes the increase in depression symptoms. So future research, just get a more comprehensive idea of all the changes that to the microbiome that can cause to this increased gut permeability. I gave one example, but I'm sure there's more changes that can cause this increased gut permeability. And then a comprehensive study of multiple probiotics and prebiotics to kind of determine what would be the best treatment option, either for preventative or for actual treatment options of mental health using the microbiome. So yeah. And also I want to give a shout out to Mike Iba for helping me and being an amazing mentor on this project.
Amazing. Let's give Karyss a resounding round of applause. Thank you so much for that wonderful talk, Karyss. We definitely have some time for questions, and I believe one just was popped into the chat. So I learned that babies with food allergy have a high chance to develop some disorders like autism. Is there some connection with the microbiome in the gut and these disorders?
Yeah, actually, that's a big thing. That's part of the reason I started looking into the microbiome. One of the chefs that I worked with at one of my previous jobs, his son had autism and he changed his diet, put him on a paleo diet, and you would not know that he had ever been diagnosed with autism before. And so the microbiome plays a huge role in a lot of different functions and or conditions in our body.
More questions? You can certainly unmute if you wanted to. Now to ask the question.
Hi, Karyss. I have a question. First off, wonderful job. I really enjoyed your presentation. I learned a lot. But my question is have you come across any ways of using these microbiomes to diagnose people with certain conditions?
I didn't actually look specifically into that, but I imagine they can, because they've found that certain bacteria are associated with higher levels of positive self-judgment, and just precursors of like more positive mental health, as well as some bacteria that are associated with increased severity of depression and anxiety and symptoms. So I imagine that they would be able to look at the composition and kind of get an idea to be able to like predict whether or not they might have some sort of mental health issue.
Sorry, Karyss, let me just go ahead and say it. So what can we do for gut permeability for bacteria? Yeah, go for it.
So for gut permeability of bacteria, so that's just, following those, or just trying to do your best to like eat a good diet that doesn't, or that does feed the beneficial microorganisms in there, exercise, things like that. I just doing the things to help to cultivate a healthy microbiome will help to decrease the likelihood of having this gut permeability. Really, I suggest eating a lot of fiber. That seems to be the big thing, like high fiber diets.
I have a question.
Yeah. So
can the prebiotics possibly feed the bad bacteria in our body as well, or does it only feed the good bacteria?
So prebiotics, they are, most of the ones that you're going to have, they're designed to feed the beneficial bacteria. Yes, theoretically they can feed the less beneficial bacteria, but they're gonna primarily feed that beneficial bacteria, and so then so that those will thrive, and eventually the non-beneficial bacteria will die out because there's just not there's not enough food for them. There's not enough, the survival of the fittest, so to speak.
Wonderful. We have time for one more question. Well, I have one, Karyss. So I guess my question is, and this could be either a really short answer, or if it's too long, just say, no, we'll talk about this later. So is it a one size fit all? Like, so I mean, I guess my impression of the microbiome, I've thought about it as more of like a signature for each individual, and no two person has the same constituency, if you will, of your microbiome. So then would treatment look like a one drug or one type of like infusion of like bacteria, if you will, or is it going to have to depend on essentially the individual's microbiome makeup?
I mean, I think that we could eventually get to a point where we could have just like a one, like fix it all, like have kind of the once we figure out the probiotics, the prebiotics, the things that really are beneficial and are going to be beneficial kind of universally. I think that we could find kind of a universal cure, but I think to start with, it'll be something more personalized because like you said, the microbiome is more of a signature to each individual.
Wonderful. Thank you so much, Karyss. Let's go ahead and give Karyss another round of applause for a wonderful talk. And we are going to keep going. Great. Okay, so our next talk is from Alex Drivas. Alex is graduating with a biochemistry and molecular biology degree and a minor in biophysics. I happen to know that Alex will be pursuing his master's in biomedical engineering at Columbia University starting this fall. And yes, Alex did work with me investigating the mechanism and chemoselectivity of HOBR-mediated oxidation of zinc-bound thiolates. Okay, Alex, take it away.
Thank you so much for the introduction, Professor Agba. Karyss, do you mind allowing me to share over my screen? Thank you. Wonderful. And can you guys all let me know if you can see the audience view? Wonderful. Awesome. So again, thank you for the introduction, Professor. My name is Alex Drivas. Thank you guys all for being here today. I will be sharing a bit about my research and how we uncovered the mechanism and chemoselectivity of HOBR-mediated oxidation of zinc-bound thiolates. Now, I would like to start off with the fact that these zinc thiolate complexes are heavily invoked in central defense mechanisms against oxidative death. They consist of a general zinc cysteine histidine architecture, where the sum of the number of cysteines and histidines equal 4. They are heavily invoked in numerous biologically relevant mechanisms, such as the chemoreceptor zinc binding domain, HSP33 molecular chaperone protein, and also yeast alcohol dehydrogenase. And while these are just a few examples of the zinc thiolate complexes, I want to share that they're all invoked in central oxidant sensing. Naturally, in response of inflammation, animal hosts, such as our own human body, produce hypohalous acids and also that of hydrogen peroxide. And we generally produce these oxidants to kill bacteria that are invading our host cells. However, these zinc thiolates present the unique ability to portray a wide variety of mechanisms after oxidizing the thiolate of these zinc-bound complexes to invoke, first, a reduction in general oxidant concentration of these heavily toxic oxidants, also a chemotactic response towards sites or away sites of inflammation, allowing them to navigate around these toxic oxidants, as well as further reversible disulfide bonds, which can lead to additional protein fold and other protein activation cascades. So my research specifically is going to be looking at the oxidation with that of HOBR, hyperbromosis. And I'd first like to start off with the general reactivity with HOBR and that of free thiols. So as I've shared, HOBR is capable of destroying a variety of microorganisms. While the concentration of chloride ions in the human host is 0.1 molar, the concentration of bromide is much lower at 0.001 molar, but this concentration is still high enough to kill bacteria. It's shown to react very, very rapidly and efficiently with thiols. And naturally, hyperbromous acid is used as a general cleaning product for many foods, such as cleaning lettuce, cleaning meat, and also used to clean our own swimming pools. So to first start off with the general reactivity with that of free thiols, I want to bring up the opportunity of reacting a hyperbromous acid with that of a central cysteine thiolate. A thiolate is just a central cysteine with a deprotonated sulfur atom, giving it its negative charge and decreasing its overall pKa. So in general, when we react a free thiol with that of our hypohalous acid, we can lead through a OH transfer or that of a Br transfer. Through this mechanism, we generally produce our sulfinic acid product, and through additional disulfide bridging, we can lead to our disulfide bond formation. Now, to speak about this OH transfer and Br transfer mechanisms a little bit more deeply, I will go into the actual mechanism. So a direct OH transfer proceeds through a nucleophilic attack of the deprotonated sulfur to our hydroxide, cleaving off our bromide in the concerted mechanism. This SN2 process produces our sulfinic acid and leaves our bromide ion as a product as well. We also have our bromide transfer, where the bromide transfer is supported by a sulfinobromide intermediate. In this case, there's a nucleophilic attack of the sulfinic thiolate onto the bromide. And in this case, our hydroxide is cleaved, placing in a negative charge and allowing it to react back with the thiol, cleaving off bromide and again producing our sulfinic acid. And what I want to take away from these two processes here is that literature invokes that the bromide transfer is more reactive over the direct OH transfer in that of free thiols. However, we want to bring up the question with that of these zinc thiolates. We know that zinc will act as a Lewis base and further decrease the pKa of that of the thiolate. So our first question we want to ask is, how does zinc affect the thiolate and hypohalous acid oxidation reactivity? And also where our work wants to go is there a unified reactivity model for predicting oxidant scenting at these biologically relevant thiolate complexes? So my work will specifically be looking at the chemoreceptor zinc binding domain, that with a single cysteine, and reacting with HOBr. This model system will allow us to specifically investigate the reactivity with a single cysteine and presents a great model to proceed with. So as I said, I'll be reacting with HOBr. We'll be able to form our products, the zinc-bound bromide, and also that of our sulfinic acid. So when we first raised these questions with Professor Agba, it was at the start of the pandemic, and we were forced to leave school. But these questions kept me up that night and continued to bug me. So I wanted to solve them the only way I knew how, and that's through computational chemistry. So to go into this a little bit more further, I want to introduce what our zinc thiolate model will be. Now, our zinc thiolate model is heavily important because it's giving us the idea of what we're reacting in our system. Naturally, in our computational model, we can't produce relative energies and accurate geometries to the extent we want to understand the chemoreactivity of that of the entire protein. So what we'll do is that we'll obtain the central active site of this thiol oxidation. So here on the left, I have the ZCB crystal structure from that of E. coli, and we can see the central zinc atom shown in purple, ligated to 3 histidine residues, and also that of our single cysteine. A 2D structure is drawn below. And to create our model system, we will be extracting this, we'll be basing off of this crystal structure to form our general model system, our optimized model system. And the goal of creating this model system is 1, to include all relevant atoms involved in the oxidative mechanism, and 2, produce a geometry and include a relevant level of theory and basis sets so that when we optimize these geometries, they're relevant and they are consistent with that of the crystal structures. So what we've done here in our model system is that we removed our 3 histidine residues and we replaced them with the midazoles, and then we removed our cysteine residue and replaced it with a methane thiol. But upon optimization, we can see that we have consistencies between our general bond lengths, between that of the zinc to the nitrogen, our midazoles, and of our zinc to our thiol in the methane thiol. So now I'll briefly move on to our computational methods. We'll start with this workflow by hypothesizing the reaction ground states by considering that of the oxidation of free thiols and that of zinc thiolates. Then we'll move into molecular mechanics calculations, where we can use Maestro Schrodinger, a program to run Newtonian mechanical force fields and run an advanced conformational searches on these ground state molecules. Now, effectively, what we want to do is produce an abundant amount of conformars by changing geometry between bond lengths and distortion and dihedral angles at a minimized geometric cost. Then we'll take these conformers and run them through quantum mechanical calculations. We'll perform optimizations on each geometry and obtain a much more accurate geometry on these conformers at a much higher level of theory and also energies. We'll take these energies and we'll extract these geometries and we'll perform numerous geometric and energetic analyses to obtain the lowest energy conformer. We'll proceed after obtaining our ground states to locate our transition states by returning back to Maestro, building transition state-like structures by performing conformationally constrained searches, or perform a potential energy surface scan between bond distances involved in elementary step, being bond broke or being formed. We'll perform a transition state optimization and follow this up with an intrinsic reaction coordinate so that we can actually plot which ground states and which stationary points the transition state connects to. Following this, we can subject our calculations to even higher energy calculations under MO66311 plus GDP and SSD for zinc level of theory and basis sets so that we can obtain even more accurate energies for these calculations. Taking all of this, we can construct our reaction coordinate and understand the actual operative mechanisms between that of our expected OH transfer, BR transfer, and determine the cumulative selectivity for each. And then what I'll be speaking about as well today is an additional analysis on comparing and trying to explain the general selectivity that we see through distortion and interaction analyses. So without further ado, I'd like to briefly introduce the mechanism of oxidation for the HOBR-mediated zinc-bound thiolate. So here we have a free energy diagram. At our 0-point energy, we have that of complex 1, which is shown as our 2D drawing of our general free thiolate. We first proceed through our ion molecule complex, where the HOBR becomes coordinated to our thiolate via an OHS interaction. Following this, we proceed through what is known as our OH transfer transition state, shown in blue, where the hydroxide becomes a transfer group to the thiolate, and our bromide is then cleaved and acts as our leaving group. This leads us to complex 4, where we have found a O-ligated zinc-bound complex. And finally, after proceeding through transition state 5, we also arrive to our dissociated zinc, sulfonic acid and our zinc-bound bromide. So I've shared here the lowest energy mechanism for that of the OH transfer. Now, I would also like to share what we've discovered for the halogen transfer, and I want us to keep in mind where the highest energy levels for our rate-limiting transition states are, because this will give us the information we need to determine the general selectivity for them. So for our halogen transfer shown in purple, we have the coordination and a general rotation to form an interaction between the bromide and that of our halogen and that of our thiolate. We then proceed through TS8, which is going to be our halogen transfer transition state, where the bromide is acting as our transfer group with the thiolate and our hydroxide is our leaving group. We then proceed to step 9, complex 9, where we have our sulfonic bromide intermediate, just like that we saw in our free thiolate, consistent with free thiol oxidation, and our zinc-bound hydroxide. Through some isomerization, we then have a sulfur-oxygen coordination, which then leads through transition state 11, finally back to complex 4, and we return to our O-ligated zinc-bound complex. That will then proceed through transition state 5, and again 6 to form our sulfonic acid product. Now, what I really want to take away from this reaction complex is the change in Gibbs free energy between transition state 8 and transition state 3. And while the oxidation can proceed through either an OH transfer or a halogen transfer, as we've shown here on this reaction coordinate, the OH transfer is energetically favored by 6.4 kcal per mole. This will lead to the general selectivity for the OH transfer over the halogen transfer. And this is what's shown with this zinc complex that's inconsistent with that of our 3-thiol oxidation. Now, we have also run the same mechanism and performed the same geometric optimizations on that of the HOCl-mediated oxidation of zinc-bound thiolates. Here we can see the OH transfer for HOCl-mediated oxidation, and we can see our Cl transfer or the halogen transfer shown in purple. Again, we've found the exact same ground states for our molecules, and we also see this consistency where the OH transfer is selected over the halogen transfer because of the difference in energy of the rate-limiting transition states, TS3 and TS8. And now that we've understood this from the general reaction diagram, I wanted to uncover geometrically and energetically why we see these differences arise, specifically in TS3 for the OH transfer and that of TS8 for the halogen transfer. So we can take a look at the actual transition states. These are geometries and energies that we've optimized for the transfer transition states of the OH transfer. Now, I want to note some consistencies between that of the HOBR system and that of the HOCl system shown on the right. First, we see the general hydroxide being our transfer group interacting with both the zinc and also that of the thiolate. And we see our halogen, whether it be the bromide or whether it be the chloride, further interacting with the imidazoles and receiving further stabilization from the complex. I would also like to note, and for us to pay attention on the zinc to thiolate bond, that of a distance of 2.4 consistent in both these structures. Now I would like to bring up the halogen transfer. And as we see on the right, what we know is that the halogen transfer experienced much greater free energy barrier to achieve their transition states. So let's look at the actual coordination between these complexes and try to understand why. So again, for these halogen transfers, we see that the halogen, respectively, the bromide and the chloride, is the transfer group in this complex. And again, our hydroxide is now our leaving group and further stable by the imidazoles. And what I'd like to specifically note is the massive elongation that the zinc thiolate bond needs to undergo to obtain this halogen transfer transition state. And this is consistent in both of the systems. This is what we believe is attributing to the chemoselectivity of that of the halogen transfer, of the OH transfer over the halogen transfer. And what's really interesting to note is that this is a complete new mechanism by introducing this zinc thiolate bond to the oxidation of free thiols that invokes this chemoselectivity for the OH over the halogen transfer compared to that of free thiols. So now, to uncover the origins for this chemoselectivity, I wanted to further explore where the components of these energy barriers are coming from in the transition state complexes. And there's a certain technique for understanding the features of the transition states. This is known as distortion interaction models. And first, I would like to bring up that for a reaction to occur, both species must distort to reach the transition state geometry. Here on the left, we have the zinc thiolate reactant of the OH transfer, distorting and requiring energy to reach its general geometry for that of the OH transfer transition state. So this will incur some amount of energy cost. So we can plot that on a general free energy diagram. We can plot this distortion and activation strain of 1. Now, this will also occur for our second reactant, that of HOBR. It will also distort to reach its transition state geometry, and this will also incur some kind of distortion energy. Now, when these species react in a transition state complex, they will also engage in stabilizing interactions that stabilize and reduce the amount of energy required for this complex. This is known as our interaction energy and can also be included on this general diagram. This is known as our interaction energy, and it reduces the overall energy and the activation energy needed. The sum of the distortion energy and that of the interaction energy are known as our general activation energy to reach this complex. So here I've shown the general distortion interaction model for that of the OH transfer. We can do the same thing for the BR transfer in that of our halogen transfer system. And we've done that. And what I specifically want to note here is the difference between the distortion that must undergo the halogen transfer compared to that of the OH transfer. And specifically in red, we can see that the zinc thiolate complex experiences much greater distortion along the halogen transfer. This is what is attributing to the general chemoselectivity that we're seeing as well. So we also wanted to do this for that of the Cl transfer. And I want to highlight, we see the same relationship occur, where again, compared to the OH transfer, the Cl transfer must undergo much greater distortion of the zinc thiolate complex to receive that halogen as its transfer group. And this leads us to hypothesize and really conclude that the preference for the OH transfer over the halogen transfer is much more strongly governed by minimizing the geometric strain of the overall complex. And again, this is not included, not incorporated in the oxidation of free thiols because the oxidation of free thiols does not include that zinc thiolate bond. So just to conclude a few central takeaways. One, while the halogen transfer is preferred as oxidation of free thiols, our work shows that the OH transfer is preferred for the oxidation of zinc thiolate complexes. Also, the zinc thiolate complex seems much more pre-organized to receive that OH transfer and doesn't require much elongation of its zinc thiolate bond, where the halogen transfer must undergo significant distortion and incur significant energetic costs to receive that halogen. And finally, this is supported by our general hypothesis and our reactivity model that the preference for OH transfer over halogen transfer is strongly governed by minimizing strain. Now, the final thing I would like to leave you all with here today is, although these SN2 mechanisms that we explored are concerted mechanisms, this research effort is not. We also have 2 additional students working on a similar project, that of Dylan Arrazati, who's working on the HSP molecular chaperone protein, and Hana Sandoval, who started working on yeast alcohol dehydrogenase. These zinc thiolates, we're also reacting with oxidants, and we're trying to further understand if we can hold with our general hypothesis for the preference of OH transfer over halogen transfer by minimizing the general geometric strain. And with that, I'd like to say thank you very much to Dr. Agba for being my PI and supporting me through this wonderful investigation, providing me all the tools to explore and become a researcher here at Chapman. I would also like to thank the additional members of the OMO research group and CUE. And with that, I'd like to open up to any questions. Thank you all for your time.
Great. Let's go ahead and give out a round of applause for a fantastic presentation. Thank you. We're right on time, but maybe we can squeeze in one question, given that I think Robby wouldn't mind a little bit of a snag. Any question for Alex that he can quickly answer? If not, we can certainly have time at the break to answer questions. Okay, well, Alex, if you don't mind, if maybe at 11 a.m. you can stick around if somebody has some questions for you, but we're going to be conscious of time. Let's give a round of applause again for a great talk. And we're going to move on to Robby. So Robby hold on, let me get my notes. Robby is also obviously graduating this semester with a biochemistry and molecular biology degree, as well as a minor in biophysics. I sense a little bit of a trend here between Alex and Robby. And Robby has worked with me investigating what makes tyrosine kinase inhibitors specific. And so Robby, go ahead and take it away.
Wonderful. Thank you for that intro. I will set up my presentation. Let's see here. Screen. And then from beginning, and of course, it's always the wrong way. Wonderful. And then before I forget, now it's all colorful. All right, hello and welcome to my presentation. Today I hope to answer or at least give you insight as to what makes a tyrosine kinase inhibitor specific. So let's start at the very beginning, the start of a targeted cancer therapy. And it began with a disease called chronic myelogenous leukemia, a bone marrow cancer. It's an unregulated growth of myeloid cells in bone marrow and accumulation which accumulate in the bloodstream. And it's caused by a Philadelphia chromosome, which is a translocation error between the chromosome 9 and chromosome 12, resulting in a protein being expressed, a tyrosine kinase called BCR-ABL. And what's interesting about BCR-ABL is that it's unregulatable. As ATP goes in and donates one of its phosphates to a substrate, which activates the substrate and then goes along a signal cascade, it's unregulatable because our bodies do not have a mechanism to work with this specific tyrosine kinase, and it completely, it's unregulatable. And so the question is, how can we attack BCR-ABL? How can we inhibit this system? And this question was proposed over 20 years ago and was answered by Dr. Brian Drucker and Dr. Nicholas Leiden, and they came up with imatinib, the first targeted cancer therapy. And this very small molecule goes into BCR-ABL, specifically on the ABL side of it, and blocks ATP from binding. And so in our first diagram, we have BCR-ABL here, which has the constitutively active pathway. And on the right here, we have imatinib, which goes in and stops ATP from being able to get to its binding site, thus stopping the substrate from being activated, which thus stops the signal cascade. And so we kind of have this huge explosion of what are called Zauberkugels for anti-cancer treatments, or magic bullets. And so we have imatinib up here, the very first generation, which was a proof of concept. And then over years, we've developed a better drug, such as nilotinib, which is about 20 to 30 times fold more effective. And now we have a third generation inhibitor called ponatinib. And this one inhibits all known BCR-ABL mutations. A lot of times these tyrosine kinases will undergo mutations that make them resistant to our current drugs. And so ponatinib was designed with the specific intention to inhibit all the forms of BCR-ABL. But this is not just for BCR-ABL, not just one tyrosine kinase. We've also got SRC kinase down here, which has gone through the exact same process, where we start with seracatinib in the first generation, which goes to PP1 to PP2, second generation. And now they're currently testing KX2391, a lovely name, for the inhibition of multiple different specific mutations. But let's remind ourselves, what are these all doing? What do they all have in common? They're all ATP blocking. And this is really important because one of the papers I read in core to my research is that ATP binding sites are conserved throughout kinases relatively highly. Of course, ATP is a very specific molecule, so they need to have similar binding sites. So this led rise to a multi-targeting BCR-ABL and SRC family kinase inhibitor. It's a mouthful, so we'll call it dasatinib. And what makes this one really special is that when we have a specific mutations within a cell, sometimes you have an SRC family kinase mutation as well as a BCR-ABL mutation. And this results in a drug that maybe can target both. And so on the left here, we have BCR-ABL being inhibited by dasatinib in the BCR-ABL, as well as ABL2, a different type of tyrosine kinase, relatively similar though. And then on the right here, we have the SRC family, and this one dasatinib goes into and binds to as well, relatively effectively. But let's really break this down. Let's see what I'm really interested here using good old dragons. So imatinib here is going to bind to the BCR-ABL dragon. It's an oncogenic tyrosine kinase, and it effectively gets it, at least in theory, right? But when we look at cancer and all the mutations and all the different issues, and when it comes to long-term treatment options, this is really a simplified picture that does not capture the entire scope of a cancer treatment. So up here we have a hydra, which I'm going to call the oncogenic tyrosine kinase, multiple tyrosine kinase hydra up here. And so it's got different heads for BCR-ABL, SRC, and LIN, which are different types of SRC family kinases. But down here, we also have BCR-ABL, and it can undergo its own mutations. So one of the heads is the most common form that we find within patients, but there are very specific mutations that make it resistant to imatinib treatments. So while imatinib in theory targets a tyrosine kinase and should solve the problem, it really only gets one of the heads of these hydras, which really represent the entire problem. So we've got really smart people who've developed solutions to both of these problems. We've got dasatinib, the one I showed earlier, which binds to and inhibits BCR-ABL, but also the SRC and LIN heads of these cancerous organisms, these cells. And then also we've got ponatinib down here, which deals with all the different mutations that BCR-ABL can exhibit. So it gets the T315I mutation, which is the most significant, I'd say, but it also gets this V mutation as well. And so this kind of led to my 3 research questions, which I'll combine to one in just a second. What about dasatinib allows it to bind to so many different tyrosine kinases? What about ponatinib allows it to inhibit so many BCR-ABL mutations? And additionally, why can't imatinib bind to anything else? Why is it so specific to a very, very specific BCR-ABL, the most common, but still very specific mutation? Which really leads me to my big research question, which I presented at the beginning. What properties make a tyrosine kinase inhibitor specific? And as this is part of the OMO lab, how do I answer that with computational chemistry? Because it's kind of hard to get my hands on some of these very expensive chemicals, especially as an undergraduate student. So my hypothesis, and this is very broad, is the more flexible a ligand is, the better it will be at inhibiting multiple targets. So let's go through my methods real quick. So first, basically what you've seen up until this point is my background research and ligand compilation. I started with a bunch of tyrosine kinase inhibitors and oncogenic pathways. I found relevant IC50 values to make sure that they've been tested in solution in a biological system. And then for the ones that I found reasonable crystal structures, I took those and I really ran with those throughout the whole project. And so to kind of talk a little bit about how I did conformation generation, because I was interested in the different flexibilities of these conformations, I took those tyrosine kinase inhibitors from my library. I did gas phase and water phase conformational searches, and I optimized those gas phases with quantum mechanics on the cluster to get really high accuracy conformations. So now let's go on to how I analyze the data. I have all these conformations now, but let's see how I analyze it. But before, oh, I'm sorry. But I'm going to look at two very specific molecular properties presented by Pungavanam et al. I really hope I'm pronouncing that name correctly. So radius of gyration is a really effective way at looking at the different sizes of conformations. It is the distance between the axis of rotation and the center of gravity of a rotating object. And what's key here is that this object up here on the left is rotating, and it has a specific rotational inertia that when we make a different model system where we have the sum of the mass a set distance away and that rotates, that set distance is known as the radius of gyration, and these two systems have the exact same rotational inertia. So it's really effective at giving us an idea of the size of different conformations. But we have also got polar surface area, because we really need to take a look at the polar sides of these molecules and how they change between conformations. So the technical definition here is the surface sum over all polar atoms, oxygen, nitrogen, etc., and attached hydrogen atoms. And it's really useful in drug design. It's got very specific properties that drugs have to be good, but it's also really useful at comparing the differences between polar region exposure in these tyrosine kinase inhibitors, excuse me. And so before I even get to my results, let me explain to you why my data is relevant. Why should you care, and why this is applicable to a biological system? More specifically, why are my generated conformations relevant to the crystal structure ligand conformations we find in these ATP binding sites? Or visually, why is the molecule on the left, when I generate a bunch of conformations, even comparable to the conformation found within a tyrosine kinase crystal structure? Before I get too ahead of myself, let's spoil the whole thing. Yes, they are relevant, but I didn't have to do all the heavy lifting here. Going back to my Pungavanam et al. Paper, to paraphrase what the overall sum of the paper, what I took away, if I capture the crystal structure ligand geometry, I have generated relevant conformations. And so here's 3 criteria that they propose that if I do, I have relevant conformations. So the ligand has a plethora of conformations we find in solution. It goes from a pill to the extracellular fluid, it crosses the membrane, it goes to the cytosol of the target protein. There's a whole thing to process, multiple different conformations. So if my ensemble has a few of those, its chances are we'll see those exhibited in the actual solution as this drug acts on the system. But also the conformation of the bound ligand varies in several ways. So here we have telithromycin, complex to a specific ribosome of an organism. And here we have telithromycin, again, complex to a different organism. And when I overlay them, you can see that they have very different conformations. Some parts are conserved, fair enough, but some are very different. And so this suggests that multiple conformations are really effective. If I capture those crystal structures in there, then I quite possibly have a good array of what the molecule could look like within different binding sites. And additionally, crystal structures are not perfect. So having a range might be more impactful than having very specific. I've recreated the crystal structure exactly to 1-to-1. Continuing on. So did I do that? Well, we have one issue here. So when I deal with bond orders and hydrogen, it's a little bit tricky because when I take these ligands out of the crystal structure, they're naked and I can't analyze these. So I got to put clothes on them effectively, right? And that's much better. So to solve this problem, I kind of developed 3 different ways to look at this. So I took this out, right? It's still naked, and I put clothes on it. And now I have to figure out where the hydrogens go and how these bond orders really look in order to analyze something like polar surface area, which matters on hydrogen placement, as well as radius of gyration, because there's a lot of hydrogens which would impact the center of mass, right? So when I use this algorithm, it's very instant. It's based on an inaccurate model. This is in Maestro. But then I used a predicted model, which is what Pungavanam et al. Did. And that's relatively fast. It takes about a minute to put these hydrogens on and kind of make sure the bond orders are correct and whatnot. It's relatively accurate. And then this is the fun part. I use quantum mechanics. It takes 1 to 5 days, usually up to 2 weeks. I'm sorry to all my other OMO friends who had to deal with that over the years. And it's very, very accurate. I used a very accurate model from a lovely Australian paper, actually. And so before I get even further into my data, let's look at this. So these represent the radius of gyration, a whole bunch of different conformations here with radius of gyration. This one has the highest radius of gyration, this one has the lowest, and this one's in the middle. You see as it gets more bunched up, it's more compacted, it has a lower radius of gyration. The more spread out it is, it's higher. And this is kind of true for polar surface area, more polar region exposure versus less polar region exposure. And so my question is my crystal structure's conformational properties within this range somewhere? Do they fall within this range? Do they? Well, when I did the guess, Not really. Only 47% of the crystal structures I found fell in my range, which is kind of a sad face. I don't know how relevant that is. So I'm going to continue on and use my predicted model, which is what Pungavanam et al. Did. And they found that they had 78%, or I found that I had 78% of my crystal structures within that range. So hooray. But quantum mechanics, here's the kicker, right? So when I use quantum mechanics, I currently have 84% of those crystal structures within the ranges. And if I just had a little bit more time, or perhaps I just inputted something wrong, I might be able to get even 2 more, which would up that to 86%, which is even better. Almost, just didn't have enough time, but that's okay. So my takeaway, quantum mechanics is actually better for better than molecular mechanics for finding relevant conformations. That's a really cool finding just on data analysis before I even get to my data. Now let's get to my data because that's exciting too. But I hope you believe me when I say my data is relevant now. Let's remind us of our questions. What about dasatinib allows it to bind to so many tyrosine kinases? Why can't imatinib bind anything else? And what about ponatinib allows it to inhibit so many different BCR ABL mutations? Let's look at ponatinib here, right? So there's a graph of all the different conformations I looked at, and I calculated the radius of gyration, polar surface area. Let's look at ponatinib down here on the left in the bolded areas. Ponatinib is a 3rd generation tyrosine kinase inhibitor, also a very good BCR ABL mutation inhibitor. And so my hypothesis would be relatively flexible, so it can inhibit all sorts of different mutations. But I find that's not the case, actually. I find that it has the 4th smallest radius of gyration and polar surface area range, very small polar surface area, very small standard deviation in general between them, but specifically for polar surface area. And interestingly, it had the highest radius of gyration conformations, but one of the lowest overall polar surface areas. And I suspect that this is because it was designed to inhibit a specific molecule and ignore mutations, and it's really specific. So we actually see it being relatively specific in the conformations it takes. Now we're going to go on to imatinib. What about imatinib? What makes it not be able to bind anything else? And I unfortunately don't have a concrete answer for this, but let's just remind ourselves which one it is. So the one underneath the arrow, above the arrow. 1st generation, not effective against BCR mutations and not really effective against most SRC family kinases in general. And so I said, I believe it. My hypothesis is it should have relatively low flexibility, but it was statistically indistinguishable from ponatinib's radius of gyration, and it had the smallest polar surface area range for mainland tyrosine kinase inhibitors. So it's suggesting that it does have a really small number of conformations it can take, but I'm not fully confident in that answer because it was so similar to ponatinib, which should be different theoretically because one is really good at everything and one is not very good at everything. So kind of in the middle there. Now we're going to go on to dasatinib, just kind of look at it, right? It's the purple one in the middle. 2nd generation tyrosine kinase inhibitor. It's a dual inhibitor, relatively ineffective against rare mutations like ponatinib is, but I still believe it would have a very high flexibility. I believe it would have the highest radius of gyration polar surface area range as well. The results found were that it has the largest radius of gyration polar surface area range, as well as the highest polar surface area standard deviation and the 2nd highest radius of gyration standard deviation. So very varied. It's very varied. As it was designed, and so my conclusion on this is that it was designed to inhibit multiple kinases, so it needs to be able to fit in multiple, albeit conserved, but slightly different ATP binding pockets. So let's look at my conclusions just to summarize everything real quick. So quantum mechanical methods are just as, if not more, useful to analyze tyrosine kinase inhibitors' molecular properties. That's a really cool finding that I found just on my own. Flexibility. Radius of gyration does not seem to predict a tyrosine kinase ability to inhibit multiple mutations of a tyrosine kinase here, but it might be able to suggest a reason for the ability for a tyrosine kinase inhibitor to inhibit multiple different kinases, multiple different tyrosine kinases specifically. And that's it. A big thank you to Chapman University Center of Undergraduate Excellence, Schrodinger, who I've had a lovely course with over the summer and got me really invested into this, the lovely people of the OMO lab, and Dr. Ogba himself. Thank you very much. Any questions, comments, or concerns?
Oh, well, hopefully no concerns. Let's go ahead and give Robby a round of applause for a fantastic presentation. Yeah, we have maybe a couple of minutes for questions. So maybe 1 or 2 questions if we have. Oh, yeah, Dr. Eva.
So that was really, really interesting because I work with small molecule inhibitors as well. So when you look at these different compounds, you look at their specificities for the targets that you test, is there anything in the literature about whether some of them have more off-target interaction sites? Is there much known about that? Did that inform you at all?
Yes. So it's kind of a much bigger picture question here. But when we talk about specificity, when you have like a doctor giving a patient a specific treatment, there's a whole bunch of different issues that might arise from using different tyrosine kinase inhibitors, which might cause toxicity. Like for instance, dasatinib is not very specific. So theoretically, you wouldn't want to give it to a person who doesn't have SRC mutations. You'd want to give them like ponatinib if they just have BC or ABL. And so the off-target issues might arise from a large amount of flexibility, right? They might be able to fit into healthy cells, tyrosine kinases, and cause toxicity. So there's definitely something here to be said about toxicity, but a lack of crystal structures and a lack of IC50 data on toxicity is kind of an issue. But yeah.
Thanks, that's very cool.
Thank you.
In this second half of our symposium, we have Dylan, who will be, hold on, let me get my notes up. I have lost that. Yeah. Okay, so Dylan is actually a double major in biochemistry and molecular biology, as well as in Spanish. Dylan has worked with me investigating, quote, mechanistic insights into hypochlorous acid sensing at a 4-cysteine zinc complex, which is implicated in bacterial redox sensing. Dylan, take it away.
Awesome. Let me put the slideshow. And do you guys see my laser pointer? Awesome. Okay. Wait, first. Wait, I need to. There we go. Okay. So today I'll be going into the mechanistic insights. As oh, sorry about that. It's my dog. Let me pick him up. Come here, buddy. There we go. So no more crying. Back to it. Okay, so today we're going to be going into mechanistic insights into hypochlorous acid sensing at a 4-cysteine zinc complex that is implicated in bacterial redox sensing, and I'll be using computational techniques to investigate this process. So first, what are zinc fingers? Zinc fingers were originally discovered in DNA binding domains of transcription factors, and they are usually comprised of a zinc architecture in which they are bound to histidine or cysteine in a tetrahedral geometry. And while zinc fingers are known to adopt various functions, such as being purely structural elements or mediating protein-DNA interactions, they can also act as redox switches. And redox switches are molecular tools that are involved in the regulation of many proteins, such as metabolic enzymes and transcriptional regulators. And literature has shown that these redox switches also belong to the cell defense system against oxidative stress, which is usually as a result of the release of reactive oxygen species, which in our case is HOCl. And another thing to note about redox switches is that they are often based on the oxidation of thiol groups. And usually the oxidation of these thiols can lead to the formation of disulfide bridges, which has been associated with important protein conformational changes. So now I'm going to be going into the particular zinc finger domain that I'm studying within the protein that I'm looking at, which is HSP33. And it is a chaperone protein that assists proteins in proper folding. And HSP33 is comprised of 3 specific domains. You have the linker region, you have the N-terminal domain, and lastly, you have the C-terminal domain that contains our zinc finger active site. And this zinc finger active site contains a zinc atom bound to 4 conserved cysteine residues. And this is the main active site that is important in determining how this protein in bacteria basically uses HOCl to escape oxidative cell death. So this protein can exist in 2 different states. In one state, you have the reduced and non-stress conditions where HOCl is not present. In this state, we have the zinc bound to the, or coordinated to the 4 cysteine residues, and the protein remains in its inactive and folded state. However, upon HOCl oxidative stress conditions or upon inflammation, what happens is you actually have a release of the zinc atom from the zinc finger domain and causing a formation of disulfide bridges between neighboring cysteines. And this is what actually allows the protein to unfold and expose this important linker region where key proteins of the bacteria can bind to and prevent undergoing oxidation and aggregation. And the literature has shown that HCl normally can cause aggregation of proteins, and we don't want that, or the bacteria wouldn't want that, or else it would lead it to oxidative cell death. So now that we actually have an idea of how HSP33 works, I want to show you guys my motivation for actually completing this work. So I won't be able to go into everything just because of the time crunch, but I will mainly be trying to elucidate the mechanism upon HCl oxidation. And I'm going to be comparing the oxidative pathways between a 4-cysteine system that we see right here and a 1-cysteine system. So our collaborator from the past year, Lindsay Zumwalt, she actually computed the same similar mechanistic oxidation pathway with a zinc bound to 1 cysteine and 3 histidine residues. And she was able to compute 2 different pathways, and I'll be comparing how the single pathway that I found compares to that. But how did I actually go about obtaining my reaction pathway? Well, first, I build up all the ground states of the reaction pathway in a program called Maestro. And what I do is a conformational search in molecular mechanics. And what this does is it produces all the coordinates of all possible conformational isomers that can exist for every structure. And the way I analyze these different conformations initially is I submit them for quantum mechanical optimizations to the cluster under the B3 level of theory. And I use a certain set or multiple basis sets to kind of take care of the other parameters in my system. But once I optimize these geometries of each conformation, I can extract energetic values and plot them on Excel and analyze, how the energies are changing from the lowest energy state to higher energy conformations. But I don't stop there. I also build most of these compounds or different conformations on a program called PyMol, in which I can analyze these favorable interactions that are seen in the lowest energy state, and then start building the conformations upon higher energy states and look at, okay, what favorable interactions that I saw in the lower energy structures, which ones are being, no longer shown. And that'll kind of give me an idea of what's important in the stability of these lower energy conformers. And this is also how I analyzed my transition states as well. And like I said, due to a time crunch, I won't be able to go into the necessary methods as to how I obtain these transition states, but I'm open to answering questions about that after the presentation. So here we have a depiction of my computed model system. Instead of having actual cysteine residues, I replaced them with thiolates, just a sulfur bound to a methyl. And the main takeaway of my computed model is that I have a comparison to the actual PDB crystal structure of HSP33. And while I computed my model in gas phase and not taking into account the protein environment, we see that we actually have a retention in the conformation between the actual solvent, PDB structure and what I computed in gas phase. And this conformation was seen in multiple ground states in my reaction pathway, helping me validate that what we're seeing in my computations could also possibly be seen in a solvent model. Now, moving on to my actual reaction oxidative pathway that I computed. So the way this reaction proceeds is you initially have your zinc tetrathiolate complex. And what happens after this is you have your coordination of HOCl that coordinates via the hydrogen. You have this HS interaction. Now, the pathway that proceeds from this is an OH transfer pathway in which the oxygen starts to rotate and it positions itself in a linear fashion to one of the sulfurs, in which an SN2 nucleophilic substitution reaction can occur. And then you have a formation of a zinc-bound sulfonic acid intermediate along with a dissociated chlorine ion. So as I said, I think I forgot to mention this before, but the formation of disulfide bridges needs to proceed through a sulfonic acid intermediate, which the sulfonic acid can then normally interact with another thiolate and form your disulfide bridge. So it's pretty important that I actually found this ground state, but it didn't stop there. I also was able to isolate a ground state in which the sulfonic acid was ligated via the oxygen, and that's about 1 kilocalorie per mole more stable than an S-ligated sulfonic acid. And while I haven't yet computed a transition state that can link these two ground states officially through a transition state, I still believe a linkage isomerization could occur to have this exchange between the sulfur and the oxygen. But it doesn't stop there either. I also computed a ground state in which the sulfonic acid actually dissociates from the zinc complex, and you have a chlorine bound to your zinc complex. So this structure right here was one of the main differences between my system with 4 cysteines and our collaborator system with 1 cysteine. In her system, this ground state where you have a sulfonic acid and a chlorine bound to your zinc domain, this structure was actually the lowest energy structure in her oxidative pathway. And I'll be going into the reasons later on as to why that might be the case in her system, whereas that was not the case in my system. And it was actually more favorable to have a zinc bound sulfonic acid as the lowest energy state for this reaction. And like I said, due to constraints of time, I won't be able to talk about why I was not able to isolate a Cl transfer pathway that Alex actually talked about earlier, in which you can form a sulfonyl chloride intermediate through a halogen transfer, and then to ultimately go back to a formation of a sulfonic acid. But like I said, I won't be going into that. I'm mainly going to be going into why is the OH transfer pathway favorable, and what are the differences in activation barriers for the OH transfer in a 4-cysteine system versus a 1-cysteine system. And lastly, I'll also be talking about why is the dissociation of sulfonic acid from the zinc complex not favorable. So if we're going to compare the OH transfer activation barriers between a 4-cysteine system and a 1-cysteine system, we need to look at the activation barriers. And if you look at my system, in order to get to the transition state that leads to the formation of a bound sulfonic acid, it's about 2.7 kilocalories per mole in activation energy. However, if we look at our collaborator system, in which we have the histidines that she modeled were imidazole groups, and her 1-cysteine was represented as another thiolate, we see that the activation energy barrier to actually reach the transition state is 7.4 kilocalories per mole. Now that's pretty interesting, but what are the factors that are actually causing this? Well, I want you to first look at the overall charge of my transition state and her transition state. We see that the overall charge in mine is a -2 charge, whereas in our collaborators complex, it was a +1 charge. And that plays a huge role in the nucleophilicity of the sulfur atom that needs to attack the oxygen to form that bound sulfonic acid. The sulfur is going to be more nucleophilic and more reactive in a system that has an overall negative charge, where instead of having an overall +1 charge. So that's my main hypothesis as to why these activation energy barriers are different. But another reason why I believe this activation barrier is higher in our collaborator system is that the initial zinc sulfur bond that was seen in the state before the transition state actually breaks to form a zinc oxygen bond and have an O-ligated sulfonic acid. Whereas in my system, we don't see that bond breakage and bond formation. We simply just have the formation of a sulfur oxygen bond and a dissociation of the chlorine ion to form a S-ligated sulfonic acid. But now that we understand why the OH transfer is more thermodynamically favored in my system, I want to go into the implications as to why the sulfonic acid needs to stay bound to my complex, and why that's important for the HSP33 protein and the bacteria and its survival. So here I've listed the equilibrium reactions between the zinc-bound sulfonic acid state and the dissociated sulfonic acid state, in which the chlorine ends up binding to the complex. I have the same thing down here for our collaborator. And the main thing that I want you guys to initially take note of is when you're going from a zinc-bound sulfonic acid complex in my system, the overall charge initially is a -1 charge. But if you dissociate that sulfonic acid and have a chlorine attached, you're going to a -2 charge. So you're increasing the overall negative charge of the system. You're going away from a more neutral state, and that's not going to be as favorable. Whereas if you look at our collaborators, reaction right here, the bound sulfonic acid system has an overall +2 charge. But then once the chlorine displaces that sulfonic acid, you end up having an overall +1 charge. So we're getting more closer to a zero neutral state, and that would make the reaction more favorable. But now I want to actually go into the biological implications as to why HSP33 in the bacteria needs to have a zinc-bound sulfonic acid. So when you want to form a disulfide bridge, you want this sulfonic acid to be as close in proximity to a neighboring thiolate so that it can actually do this nucleophilic substitution reaction and displace that hydroxide and form your disulfide bridge. If we were to have this this sulfonic acid dissociate from the complex, first off, it's not going to be anywhere near another one of the thiolates to form your disulfide bridge. And you're also leaving a more like variation and chance of the sulfonic acid interacting with other surrounding amino acid residues, and you're not going to get that formation of your disulfide bridge that is needed for the protein or for the bacteria to survive. Because again, the way the bacteria survives is upon HOC oxidation, you have a release of zinc and the formation of your disulfide bridges, which then unfolds the protein, exposes your linker region, and that's where the key proteins of the bacteria combine to and then prevent undergoing oxidation, aggregation, and ultimately prevent cell death. So those were my main findings, and some key takeaways of my research is that while the CL transfer pathway could not be found, we were still able to isolate a favorable OH transfer pathway that can lead to the formation of disulfide bridges. And we found that it was more thermodynamically favored to have a zinc-bound sulfonic acid instead of a dissociated sulfonic acid. And lastly, the OH transfer activation energy barrier is lower within a 4-cysteine system. But now for future directions, where do we want to go about this? Well, it just so happens that our newest member of our lab, Hana Sandoval, she is going to be computing the HOCO immediate oxidation pathway of a system that incorporates 2 cysteines and 2 histidine structures. And I want to basically see, from her research, will there be an isolatable CL transfer pathway? Just briefly, one of the main reasons why the CL transfer pathway was not viable in my system was just due to the overall electronics of my system. Having 4 cysteine residues or 4 thiolates increases the overall charge of the system. And when it's like that, it's more flexible and subject to variation of things that can happen. And there were certain favorable hydrogen bonding interactions that were occurring that prevented the CL transfer pathway from occurring. So it'll be interesting to see, like, okay, if there are still 2 cysteines that are pretty electronegative, will these other histidine amino acids play a role in stabilization for a CL transfer pathway, or will we not, see a CL transfer pathway and still see a preferred OH transfer pathway? But yeah, that's basically my key findings of my research. I'd like to acknowledge Dr. Ogba for being the best PI ever and for always giving me amazing advice, not only in school, but just in life. And I want to thank everyone in the OMO lab, especially Dr. Han and Alex Drivas. You'll see why I call him Dr. Han in a second. He's going to give you guys an amazing presentation. He really probed some amazing thesis level questions throughout this research and really taught me to think critically. And I want to also thank the entire biochem department at Chapman for just giving me an amazing, education. I've learned so much, and I know I'll be able to take my skills that I've learned into my future endeavors. Thank you.
Wonderful. Let's give Dylan a round of applause for a fantastic presentation. Thank you so much. And we have time for one question before we move on to the next. So if anybody has any pressing questions, please, the floor is yours. Well, Dylan, I'm not going to ask you a question about the research given it's our work, and I don't want to put you on the spot. But I will ask you, what are you thinking about moving in the future? What is your sort of the plans moving forward?
So this has been a huge decision, for me. And while I actually was initially recently thinking I wanted to do a master's in business and science, I've started to come back to the idea of wanting to do a PhD. I feel like I can handle it. And I know I talked to you about this and kind of building up back that confidence that that I'm able to do research. And even though there's a lot of failure that comes with it, to be actually able to pursue through those failures and ultimately get to, where you want to be in your research, and also just take that life lesson in life. You're going to fail sometimes and you might not always be doing what you ultimately want to do, but then that'll lead you to your, your final, destination of where you want to be. So I think it's important to, accept those harsh endeavors, but to get to where you want to be, So yeah, definitely a PhD, I think. So you convinced me, you took me away from the med path and got me in the PhD path.
And I agree. I told Dylan this yesterday. The way you were able to explain this to me at your poster was just absolutely amazing.
Thank you. I really appreciate that.
Well, Dylan, this is news to me and very excited. So let's definitely have a conversation after this. So
Yeah. We've got we have to have a whole gap year to figure this out.
Yes.
So we'll stay in contact.
Yes, I'm very much supportive of that. Let's thank Dylan again for a wonderful presentation. Great. Okay, let's move on. Our next presenter is Brian Han, or as his co-workers call him, Dr. Han. Brian will be, well, he's graduating with a bachelor's in biochemistry and molecular biology and a minor in economics. I don't know how you mix those two, but that's really cool. Brian actually has been working on sort of a medicinally related work, but investigating sulfur 6 fluoride activation using calcium salts. Brian, take it away.
Okay, I hope you could see the presentation. Okay, thank you for the introduction, Dr. Agba. And with the introduction of Dr. Dylan, there's much pressure on my shoulder now. I'll try my best to explain my research. Well, my research is about this, but it's one heck of a title, so I'll give you a shortened version. Sulfur fluoride bond activation using calcium salts in the synthesis of sulfonamides, which is commonly used against cardiovascular, infectious, and neurological diseases. It constitutes 15% of the top 100 most prescribed US FDA-approved drugs. A conventional way to synthesize sulfonamide includes a two-step synthesis, where the first step includes the oxidation of sulfides to produce sulfonyl chloride. But this first step requires the use of strong oxidants, and that makes this first reaction incompatible with the myriad functional groups. The second reaction involves the nucleophilic addition of an amine towards sulfonyl chloride. Because of the weak sulfur 6 chlorine bond, this reaction is highly reactive, and we can't expect much selectivity in the presence of competing nucleophiles. As an alternative to sulfonyl fluoride has been introduced to the field. And unlike sulfonyl chlorides, it has a very stable sulfur fluorine bond, and this reaction is quite unreactive. So it typically requires performing the reaction under harsh conditions, such as putting the reaction under extended heating. Back in 1930s, it was attempted to produce sulfonamide from sulfonyl fluoride, and had to be heated up to 240 degrees Celsius, and you can imagine how difficult it is to synthesize that bond to form sulfonamide. Dr. Ball in pulmonology and his collaborators were able to synthesize a variety of sulfonamides, which looks like this, by adding salt into reaction. Well, what do I really mean by salt? Am I adding a table salt that you can commonly find in dining tables and hope that the bond would be activated to form sulfonamide? Well, by definition, a salt is any chemical compound that contains a cation and an anion. So table salt that has a chemical formula of NaCl, that's not the one that we use. We're using calcium salt, and specifically calcium trifluoride that consists of calcium and 2 trifluoride anions. That's what calcium trifluoride looks like. I'm going to switch that salt to calcium trifluoride, which is also a Lewis acid. And how do we, one sec. All right, so among the many sulfonamides that our experimental collaborators have synthesized, we decided to focus on this one reaction, the synthesis of N-phenylbenzene sulfonamide from benzene sulfonyl fluoride and 2 equivalents of aniline. The reaction had not been elucidated yet, so our first research goal was to elucidate the mechanism of the sulfur 6 fluorine bond activation to form sulfonamide. This is a table obtained from our collaborators. As you can see, they tried using different types of Lewis acids. Using calcium trifluoride produced the most amount of yield. Using other group 2 metal trifluorides, less yields. And interestingly, calcium with other anions, calcium triflate, led to a significantly decreased yield of 4%. Using group 1 metal, only 2%, and without any Lewis acid, there was no reaction. So from this table came our second research question: what is the role of calcium and what is the role of trifluoride? A recent study done by our collaborators suggests that adding DAPCO and aniline and performing the reaction in THF as opposed to adding 2 anilines and performing the reaction in terramyl alcohol led to a more efficient synthesis of sulfonamide. What do I mean by that? The second reaction was complete only after 3 hours at room temperature, producing a greater yield, whereas the first reaction was complete after 24 hours and had to be heated up to 60 degrees Celsius. So from this data came our third research question: what is the role of DAPCO? Well, I'm in a computational chemistry lab, so how do we go about probing the effect of DAPCO computationally? So I decided to switch the solvent from the first reaction, from teramyl alcohol to THF, so that the only thing difference between these two reactions is the fact that the first reaction involves 2 aniline, the second involves DAPCO and aniline, while keeping everything else constant. And that way we can investigate the effect of DAPCO in the reaction. How do we go about answering other research questions? Well, this is myself thinking how an optimized geometry of terbutanol would look like. And I'm going to use terbutanol just for an example to demonstrate our computational methods. So we construct a 3D molecule, a 3D structure of terbutanol in this program called Maestro, and perform conformational search, which is based on molecular mechanics. In this step, we can obtain as many conformational isomers of terbutanol in a relatively short amount of time. And at this stage, we're obtaining geometries of those isomers that haven't reached their local minimum yet. Next step, we apply quantum mechanical calculations onto those conformational isomers obtained from the previous step, borrowing the computational powers of the cluster. And this typically takes a longer time because we're now considering the behaviors of electrons in this molecule. And at this stage, we are bringing the energies of those conformational isomers to their local minima. And after that, we can extract thermochemical values like delta G and delta H, as well as an optimized geometries of those conformational isomers. This was the steps for obtaining ground states. Obtaining transition states is similar, but just a bit more complicated. But after computing transition states and ground states, we can obtain their thermochemical values and construct a reaction coordinate diagram to probe their thermodynamics and kinetics. So when adding calcium trifluoride, the Lewis acid, we saw great yields of sulfonamide from sulfonofluoride, the unreactive sulfonofluoride. What is so special about calcium trifluoride that it's activating sulfur 6 fluorine bond? Well, let's figure that out. Let's consider these mechanisms. Is this possible? Is it possible for that fluorine to be displaced from the sulfur center to form the fluoride ion? Or is it possible for the aniline, the nucleophilic aniline, to perform a nucleophilic attack to displace a fluoride ion? But I don't think it would be possible because fluorine ion is just too unstable for bond cleavage. Fluorine atom, as you all know, is the most electronegative atom on the periodic table. So posing a negative charge on that already electron-dense atom would be just too unstable. So what do we have in our reaction flask? We have calcium trifluoride. And calcium can act as a stabilizer of the fluorine that's developing a negative charge. And prior to any reaction occurring, calcium can ligate to calcium so that calcium can ligate to fluorine, so that as a negative charge develops on the fluorine atom, calcium can act as a stabilizer through Lewis acid and base interaction. And our computed transition states see this as a requirement in both pathways. So in both reactions, the only thing difference is that the base is the aniline in the 2-aniline pathway, and DAPCO is the base in the DAPCO pathway. And we see that base deprotonates that proton and grants a negative character on that nitrogen. That makes the nucleophilic attack favorable, thereby displacing that fluoride ion. Then as that fluoride ion gets formed, calcium goes on to stabilize that negative charge. And we saw in our transition states that this happens in one step or in a concerted mechanism. So now let's take a look at the reaction coordinate diagrams. First, we're going to take a look at the reaction coordinate diagram of the 2-aniline pathway. For time's sake, I'm just going to focus on the more important theme of the reaction, kinetics. The reaction coordinate diagram starts with this calcium trifluoride complex with 4 THF molecules ligated to it. We call this the resting calcium complex because this was the lowest energy complex prior to reaction occurring. Let's examine 3D structure of the transition state. We have the aniline, nitrogen in the back, deprotonating that proton attached to the nucleophilic aniline. Then as it happens, negative character is developing on the nitrogen of the nucleophilic aniline, and that nitrogen performs a nucleophilic attack, displacing that fluoride ion. At the same time, calcium stabilizes that fluoride ion, forming our product, our final product over here in the product complex. Next, I'm going to show you the DAPCO pathway. It also starts with the same resting calcium complex. Oh, I forgot to mention that the energy barrier of the two-aniline pathway was 21.7 kilocalories per mole. The DAPCO pathway, it had to climb up an energy barrier of 17.6 kilocalories per mole. Let's examine the 3D structure of the transition state as well. Well, the only thing difference, like I said, is that the base is DAPCO. So this DAPCO in the back, the nitrogen performs a deprotonation of that proton attached to the nucleophilic aniline. And as negative charge develops, or negative character is granted, a nucleophilic attack occurs towards the sulfur center, displacing that fluoride ion, that the fluoride ion is being stabilized by calcium. So comparing the two pathways, DAPCO pathway was computationally revealed to be a kinetically favored pathway, which is consistent with the experimental data. Well, our experimental conditions of the aniline pathway suggest that this reaction had to be heated to 60 degrees Celsius. The DAPCO pathway was complete at even room temperature, which suggests that the energy barrier is lower for the DAPCO pathway. Why do we see such observation where the DAPCO pathway is kinetically favored? Well, the main reason relies on the basicity of the bases used in each reaction. We see that the pKa of the conjugate acid of DAPCO is higher than that of aniline, suggesting that the DAPCO is stronger base than aniline by a factor of 10 to the 6.5 volts. Thus, it requires less energy to deprotonate aniline. And that was seen in our computational data as well as our experimental data. So we analyzed the transition states at an atomic level. We decided to zoom out and look at the forest instead of the trees and examine the transition states from a bigger picture. A parameter that we use to see the transition state from a larger picture is dipole moment of the entire complex. Well, what is dipole moment? It's a measure of polarity, and it's a vector quantity. Examining the dipole moment of this boron trihydride molecule, it's completely nonpolar because the vectors all cancel out. Going back to our reaction, the solvent that is used in the experiment was THF. And based on the relatively low polarity of THF, we hypothesized that conformers of ground states and transition states that exert high dipole moments will be higher in energy because complexes with high dipole moments will experience repulsion in the THF solvent. In other words, high dipole moment complexes will dislike the relatively nonpolar THF solvent, which relates to the phrases that you might have come across in your chemistry lectures in the past, that like dissolves like, and polar likes polar, and nonpolar likes nonpolar. So to test our hypothesis, we generated a delta G, or relative energy versus dipole moment plots of ground states and transition states. Each of these dots in this figure represents a conformational isomer of this ground state, the initial calcium complex that I've shown you before. And we see almost no correlation between the relative energies and their dipole moments. However, we saw a positive correlation in both pathways between the relative energy and the dipole moment. We'd have to further analyze the 3D structures of the transition states in depth in relation to their dipole moments, but currently we conjecture that the TS is trying its best to reduce the dipole moment to achieve stabilization, because it is in the transition state that the complex starts to get polarized by developing a fluoride ion and a positive charge on the bases. So that was all. To give you a summary, our reaction mechanism was revealed to be a concerted mechanism where deprotonation of aniline by base, nucleophilic addition of aniline, and sulfate 6 fluorine bond cleavage all happen at the same time. What's the role of calcium? We established that calcium acts as a stabilizer of the negative charge on the fluoride ion. And why do we see a kinetic preference in the DAPCO pathway? Simply, DAPCO is a better base than aniline. And from the bigger picture, the transition state seems to be governed by the dipole moment. In the future, we plan to analyze the 3D structures of transition states in relation to their dipole moments, and also figure out the role of trifluid. I'd like to send great gratitude to our principal investigator, Dr. Ogba, as well as our fellow research members and the NIH for funding our research last year during the summer and fall. Well, thank you for listening to my presentation, and I'll be open to any questions.
Let's give Brian a round of applause. Fantastic presentation. We certainly have about 5 minutes, so we can have a couple of questions. So please take it away. You can unmute or put it in the chat box.
I was trying to, if I may just jump in, it's a fantastic presentation, Brian. Very impressed. I was trying to follow this as a biochemist. Can you go back to that statement you made, polar likes polar and non-polar likes non-polar? What was that all about again in a phrase?
Okay, so polar molecules are more likely to form favorable interactions with other polar molecules. And when, for example, when we put an oil drop billet in a flask that contains water, we see like repulsions going on throughout, like separation of the layer, right? And that separation of layer is an unfavorable interaction. So that's what I want to point out.
Got it. I think I got it. And the non-polar is, of course, the hydrophobic forces, right?
Right.
Under interactions. Thank you.
Got time for 1 or 2 more questions.
I just wanted to give a verbal shout out to those chemistry animations. Those are prime.
Thank you.
Okay, I'm going to ask a question which is a little different. So, when you do the computational chemistry and everything else, now you end up, what if you had to pick one thing you'd like to test in a lab from all of this, what would it be? I mean, I don't mean a computational lab, I mean like a wet lab. What would be the one experiment you would take to test your model?
Related to, related to our reaction of interest, right?
Yep, exactly, yeah.
So there was one uncertainty. So I'll go back to the slide. Okay, so our, whoops. I clicked too many times. Yeah, opening Zoom and presentation at the same time takes a lot of memory, I think. Okay, going back to this slide. So in this presentation, we focused on the effect of DAPCO because we saw that the difference between these two reactions is the fact that the second reaction contains DAPCO, but the first doesn't contain aniline.
Yep.
Another thing I would like to test is, well, we use different solvents as well. Like what role is a solvent playing? That can be simply rectified if we perform the reaction, like perform the second reaction using tert-amyl alcohol. We can examine if solvent is playing a role in making like the reaction in DAPCO more efficient. If that makes sense.
Yeah, no, that's, so yeah, I get that. That's a very, I have to say, that's a really, really good answer, because that's exactly it. You give yourself more data for the rest of the work then. Yeah, I get that completely. That's great. Thank you.
No problem. Thank you.
And with the 1 more minute, Brian, any future plans you want to share with the group?
Sure. So I haven't decided permanently on what my career path will be, but I'm thinking between the choices of applying to PhD programs or going to medical school. So after graduation during the summer, I was planning to do clinical experiences to really figure out if that is my pathway to go, because I don't want to regret after like getting accepted, if I get accepted to medical school. Yeah.
Wonderful, wonderful. Well, I have no doubt that regardless of what you choose, you are going to be successful in your endeavor. So let's go ahead and give Brian another round of applause for a fantastic presentation. And, and we're just in time, and I'm sure all of you are probably done with computational work, and that is fine. Our next set of work are going to be slightly different turn of events, but Hana is going to take it away. Hana is a double major in biochemistry and molecular biology, as well as in health sciences. And I think you would see that her work actually kind of spans both of these entire colleges. Hana has worked with Dr. Jason Douglas over in the health sciences, investigating fundamental causes of racial and ethnic COVID-19 related health disparities. And obviously very relevant to where we are right now. So Hana, go ahead and take it away.
Hi, thank you for that introduction. Let me get this all set up.
Oh, sorry.
Can you guys see the presentation?
Yes.
Okay, awesome. Okay, hi everyone, my name is Hana Neutz, and for my capstone, I wrote an extensive literature review entitled Fundamental Causes of Racial and Ethnic COVID-19 Related Health Disparities. While I know COVID-19 has kind of been our world for over the past year, I'm going to give us a little background to get us all on the same page. So the SARS-CoV-2 virus created the COVID-19 pandemic. And it's infected 156 million people and killed 3 million, with this number really increasing every day. And what's something that's less well known about this is that it affects people of color in the US at much greater rates. So Black people make up 12.4% of the population, but make up 14.9% of the deaths. And if we can go over here and we can see this in a different way. So if you take the US population, 178 out of every 100,000 people that are Black die from COVID-19, while 124 of white people out of every 100,000 die. And when this is scaled up to the population of the US, the difference between 178 and 124 is a very large number. And this clearly depicts the racial disparities associated with COVID-19. So my research question was, what is the root causes of these racial and ethnic disparities, both in morbidity, which is cases of COVID-19, and in mortality, deaths from COVID-19. And my initial research on this topic gave me two vastly different explanations, one on the cellular side and one on the systemic racism side. And I found no papers that looked at both when I did my research. So my aim with this paper was to fill the gap in knowledge and use more of an interdisciplinary approach to examine what was causing these disparities. So to answer my research question, I did a literature review of 65 peer-reviewed sources, and I found these sources using Google Scholar and Chapman Leatherby Library databases, and these are some of the keywords that I used in my research. So hypertension or high blood pressure is a comorbidity factor of COVID-19. And what this means is that hypertension makes COVID-19 symptoms more severe, usually resulting in hospitalization or being put on a ventilator. Now, how this connects back to my thesis is that 46% of white Americans have hypertension, while 56% of Black Americans have hypertension. So this could be one of the factors that increases why Black people are dying at higher rates than white people due to COVID-19. But what is causing these hypertension disparities? So looking at COVID-19 disparities through this hypertension lens, there were two main pathways that emerged. First, I looked at how Black people experience systemic racism, which perpetuates poor health through a plethora of pathways, including chronic stress, and that's leading to more COVID-19 cases. And the second pathway I explored was through the cellular connections. I knew that many of the drugs that were used to cure hypertension acted on the same receptor that COVID-19 uses to gain access to our cells. So I knew there had to be some kind of connection there. And by looking at these two perspectives, I tried to answer the question of why these disparities were occurring. So diving a little more into the systemic racism literature, I found that systemic racism is really deeply ingrained in our society, and it affects populations of color in two main ways. It causes them more chronic stress and it causes them to live in lower income communities at higher rates, and this is because of residential racism. Both of these lower overall health, which leads to more COVID-19 infections. So when someone is stressed, your body exhibits a physiological response to it. So, and that physiological response is a raise in blood pressure. So chronic stress causes hypertension, which causes more morbidity associated with COVID. And this is just one aspect of it. More stress also causes worse health behaviors, which are used to compensate with that stress. And examples of this are higher rates of smoking and alcohol abuse in populations of color. And these negative health behaviors have a negative effect on your health, making you more susceptible to COVID. Chronic stress has also been directly correlated with lowering your immune system, which makes you more susceptible to COVID. So an example of this would be getting sick right after finals. You know, the stress that you're put under suppresses your immune system and makes you more susceptible to colds and infectious diseases. So now systemic racism also leads to residential segregation, which is partially attributed to the fact that redlining was allowed until 1968, and that gave banks the power to reject loans to in certain areas, causing people of color to live in lower income communities. And these low income communities had less healthcare. Just in general, lower income communities have poor healthcare services and less resources in these healthcare, in the healthcare industry. So for example, my research advisor just did a study in New York about how low income communities have less hospital beds for COVID-19 patients, even though they have way more COVID cases. And so these less healthcare services lead to more people dying of COVID-19 and it hitting that population harder. So also low income communities experience neighborhood disadvantage, which is can be through environmental racism, which has more, lower income communities have like more polluted air and dirt and just more unhealthy living conditions. And they also live in closer proximity, which enhances the spread of the virus. Lower income communities also have lower quality education because schools are funded by property taxes. And these property taxes, the homes are less expensive, so they get less funding for their education system. And education is so important for health because it actually is the #1 predictor of health and life expectancy. So you get lower education or less education, you have lower health, and your chances of getting COVID are much higher. So overall, my findings found that systemic racism is a cause for health disparities in COVID-19. So now to get a little deeper look at the cellular connection. So the ACE2 receptor found in your is found in cells all over your body. And here it is on the right in blue with its Docker protein, BOAT1. ACE2's main function in the system is to actually regulate blood pressure as part of the angiotensin system. But it's the same receptor that SARS-CoV-2 uses to gain access to your cells. So here on the right is, this is the spike protein, which is the crown-like projections that are on the coronavirus binding to the ACE2 receptor. So I thought same receptor, you know, there's got to be a connection. And So ACE2 is a part of the angiotensin system, as I said earlier, and it's a little complicated, so stick with me. But angiotensin 2 is a hormone that increases blood pressure. And ACE2 is the receptor that converts this angiotensin 2 to angiotensin 1,7, which lowers blood pressure. And ACE-I and ARBs are two classes of hypertensive drugs, both lowering blood pressure. The ACE-Is stop the production of angiotensin 2, which lowers your blood pressure. And ARBs stop angiotensin 2 from binding to its receptor to exert its effect. Now, previous studies have shown that ACE-Is and ARBs not only improve the ACE2 receptors abilities, but they also produce more ACE2 receptors. And this could be detrimental in a COVID-19 infection, right? So as there are more ACE2 receptors, it could mean a higher chance of being infected, right? But so back in July, when I started reading this literature, this is all that was out there. There were no studies about whether this was true or not, and whether this was causing the disparity between races. And this is really what drove me to want to study this in the first place. But as the months went on, there were 3 very important papers that came out in October and November of 2020. First was an in vitro experiment that said, yes, these drugs do increase ACE2, the amount of receptors, but it wasn't in the respiratory system where COVID-19 infected. It was only in other parts of the body. The second and third were population statistics studies that found there were no increased risk of mortality and no increased risk of morbidity when using these drugs. So no increased risk of getting a positive COVID-19 test. So these 3 studies kind of changed the course of my research and ruled out the cellular argument as a cause of racial disparities. So in conclusion, these racial health disparities do not stem from a cellular connection between hypertension and increased risk of COVID-19 contraction. Instead, my research found that these morbidity and mortality disparities are a result of systemic racism. And it's important to note that there could be many other factors playing a role in this. There always needs to be more research done on this subject, but hopefully this starts to bridge the gap between the 2 fields of science. Now, focus really needs to be shifted to how we can implement this, to research more on inequality and systemic racism and how it's caused a disparity in this pandemic. And we really need to focus on enacting policies that will provide equitable access to education and work towards improving health outcomes in these low-income communities and communities of color. Thank you to my mentor, Jason Douglas, and his research group. And I'd also like to thank Dr. Owens for helping me with my structural analysis. And thank you all for being here today. I will now open it up to questions.
Amazing. Let's give Hana a round of applause for a fantastic presentation. It's cool to see a very multidisciplinary effort to try to understand and this very important topic. Let's go ahead and open it up for questions. We have quite a bit of time, so certainly if you have any questions, please do not hesitate to take it away.
So can I thought this was just really fascinating. So do you think that what you've got here is such a sort of defined data set that you're really seeing something that's reflected more broadly in healthcare in general, and that this is just, you're able to see this more clearly because you've got such defined outcomes and symptoms and everything else through this?
Yeah, I think my goal was really to look at it in multiple angles, because there are a lot of factors playing a role here, in this. And yeah, I really found a very strong connection with the with the systemic racism. And so, yeah, I think by looking at it at multiple angles and kind of starting to rule things out, you know, you can have a more defined saying, yes, this really is contributing to causing these disparities.
So, and then this presumably leads to a bigger road plan, not just for the pandemic, but more broadly in healthcare in general then?
Yes, definitely. Targeting, you know, changing the education system to be, you know, to be funded more equitably and having more education for lower income communities and higher education at that will start to even out the health disparities that are in our country.
Yeah, this was great. Thank you.
Thank you.
Hana, this is Dr. J. Wonderful presentation. Really enjoyed it. So sort of dovetailing your comment regarding the education system, which you know, we know full well, right? Education is one of the top predictors of health outcomes. And that's a long-term solution. And I'm on board. We should begin to advocate for education reform. In the meantime, though, you know, take this early pandemic example of, you know, folks being triaged on sidewalks outside of hospitals in New York City last year, April. You know, sure, we could have used more resources in those communities to address the pandemic at that time. Do you know, were those resources, could they have been put in place effectively within communities that needed them most?
Yeah, I mean, we definitely had the ability to do it, and we had the resource, and we kind of just, we put them in the wrong spot. And so I think research in this realm, you know, really increasing the hospital beds in the low income communities would have helped, you know, shift the way that this pandemic panned out for the populations of color. So I think having scientists play a larger role in the policy making, you know, with these types of findings would be very, very beneficial for the health of our communities and our lower income populations.
Very good. So would you then advocate for sort of a more of a two-pronged approach, sort of, shall we say, mitigation regarding education policy? So this is sort of fixing the root causes of the problem while simultaneously advocating for adaptation and community capacity for bringing the resources that we need to sort of at least put a Band-Aid on these challenges while we're dealing with education reform.
Yes, definitely. I mean, I think we need to tackle it from multiple angles, and not just that, but also tackling systemic racism, just the biases and lowering the chronic stress that is placed on our populations of color. And, you know, that's the ultimate goal, but it's obviously very hard to just say, you know, we need to change this, but we need to create actual steps and a way to implement these changes in our society. So I would say it's more than a two-prong approach, it's a multi-approach.
Wonderful, Hana. I'll take the liberty of giving the last question. I'm so curious with your, you know, you've really amassed quite a large span of knowledge and experience at Chapman. I'm sort of wondering where you're planning on taking your career towards in the future, if you don't mind sharing with us.
Yeah, I'm extremely interested in public health, and that's kind of the direction I decided to go. I do love biochemistry and, you know, kind of the laboratory sciences, but I want to use that background and my background in research to really work either towards global health or public health policy. So I'm planning on taking a couple of years off and getting some work experience, and then going to get my MPH or possibly a PhD, but we'll see.
That's wonderful. That's wonderful. It sounds like you're preparing very nicely for that field. So let's go ahead and give Hana another round of applause for a fantastic presentation. Thank you so much. And we are going to move forward. We have Desmond, Talia. Desmond is actually a graduated senior major in biochemistry and molecular biology and a minor in psychology. And Desmond worked with Dr. Montazeri, investigating CRISPR/Cas9 delivery and genome editing. And so once again, another very relevant and on everybody's mind topic. So Desmond, please go ahead and take it away.
Okay. Can everyone see my screen? Okay, thank you for the introduction, Dr. Ogba. So my capstone's research is on CRISPR-Cas9 delivery and genome editing, specifically looking at CRISPR-Cas9 to knock out expression of the RPS6-KA5 gene in breast cancer cells. So first, I just want to give you guys a little outline of what I'll be speaking about today. I'll give an introduction on what CRISPR-Cas9 is, its components, and mechanism of action. Then I will talk about the different Cas9 delivery methods, and then I will go over the delivery systems, cell lines, and analytical methods that we use for this experiment. Then I'll talk about some of the results we obtained so far and give some future directions for where we can take this project. So what is CRISPR-Cas9? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, and it was discovered as a defense mechanism in bacteria to fight off against invaders. So CRISPR has two components, the CRISPR-associated protein, or Cas9, which can be seen here in blue, and the guide RNA, which is here in orange. So the Cas9 protein is an RNA-guided endonuclease that will create double-stranded breaks upstream of the protospacer adjacent motif seen here. So if we look at the protospacer adjacent motif, right upstream is where the Cas9 will create these double-stranded breaks. As for the guide RNA, it has two components, the CRISPR RNA or cRNA and transactivating CRISPR RNA or tracker RNA. And essentially, this guide RNA can be synthetically replaced by single guide RNA or sgRNA, which acts as, it essentially contains the CRISPR RNA tracker RNA duplex and acts as a chimera. So following Cas9 cleavage, there are two repair pathways that can be taken by the cell, either non-homologous end joining or homology-directed repair. Non-homologous end joining is an error-prone repair pathway because it doesn't use a DNA template to repair the lesion. So there are insertions and deletions that are created to essentially knock out gene expression. On the other hand, there is homology-directed repair, where the cell will utilize homologous templates from a sister chromatid to knock in gene expression. So here are the different Cas9 knockout approaches. There's plasmid DNA, mRNA, and protein. For plasmid DNA, it is a cost-effective option, but it has the slowest onset of gene editing activity seen throughout these three different approaches. mRNA is a faster method of gene editing efficiency, and it eliminates the risk of insertional mutagenesis. Ribonucleoproteins, or RNPs, offer the most rapid pulse of gene editing out of all three approaches. So just to dive in a little deeper with plasmid DNA, the components are Cas9, the CRISPR RNA tracker RNA duplex, ampicillin resistance gene, and markers, which are either a fluorescent label or an mRNA that is expressed on cell membrane proteins. Some advantages to plasmid DNA delivery is that it's a cost-effective option because the only setup required is plasmid preparation, and it also yields the longest expression time in the cells. However, it does come with some disadvantages in that it's the slowest onset of gene editing when compared to mRNA and protein delivery, because transcription and translation are required for the synthesis of the Cas9 protein. Also, there's another disadvantage through the risk of insertional mutagenesis, which is essentially a mutation on the DNA strand by addition of one or more base pairs. It also has sustained expression of Cas9 that may increase the chances of off-target cleavage events seen throughout the cell. For CRISPR delivery via mRNA, the components are the RNA for Cas9 and guide RNA. The advantages is that it has faster onset of gene editing when compared to plasmid DNA because transcription is no longer a requirement. It also reduces the chances of off-target cleavage events. However, there are some disadvantages. mRNA is highly unstable, and this creates some challenges in terms of protecting the RNA. And also, it has transient Cas9 expression in the cells. For CRISPR delivery via ribonucleoproteins, or RNPs, the only components are the Cas9 guide RNA complex. The advantages is that it does not rely on the cell to make the Cas9 protein, and that it has immediate gene editing in the nucleus because the complex is already in the form needed for genome editing activity. It also has higher gene editing efficiency compared to plasmid DNA and mRNA, and has minimal off-target cleavage events. However, there are some disadvantages that come with using RNPs. It is very challenging to deliver the RNP complex to the cells, and there is an added risk of bacterial endotoxin carryover and triggering of serious immunological responses, which can be seen through human clinical trials. So for our experimental approaches, our delivery systems are lipofectamine, peptide lipid-associated nucleic acids, or Plan A's, cationic polymers, and hybrid peptides. Lipofectamine and cationic polymers are commercially made transfection agents that we used, and Plan A's and hybrid peptides were designed and created in one of Dr. Montazeri's labs. For the cells we use triple-negative breast cancer cell lines, or MDA, MB231, and human embryonic kidney cells, or HEC293. For analytical methods, we use fluorescent microscopy, confocal microscopy, and T7 enzyme digestion. So fluorescent microscopy and confocal microscopy are pretty similar in that they both use a fluorescent microscope to study the properties of organic and inorganic substances. However, confocal microscopy offers better overall quality images because it is a 3D rendering, and you can basically track exactly where the fluorescent signal is within the cells. T7 enzyme digestion is a mismatch assay that we use, and this assay allows for quick qualitative assessment of gene editing efficiency in our CRISPR-treated cells. And how this enzyme works is that it will cleave mismatched DNA in our CRISPR-treated samples. And we know this, or the T7 enzyme will cleave the heteroduplexes that we see within our targeted DNA. So to demonstrate efficient plasmid delivery, we exposed our HEC293 cells with plasmids expressing green fluorescent protein. And we evaluated protein expression as a successful sign of transfection by using fluorescent microscopy. So here are the different delivery systems that we use. We use lipofectamine, and these 3 peptides are hybrid peptides. Unfortunately, I couldn't disclose the names of it because they are patented by Dr. Montazeri's lab. So here in lipofectamine, we show that it is clearly efficient in terms of plasmid delivery to our HEK293 cells. However, we do see signs of toxicity, which can be seen here in this region due to clumping of the cells. Our hybrid peptides also show varying degrees of efficacy in terms of plasmid delivery to our HEK293 cells. So we also delivered our CRISPR-Cas9 components as ribonucleoproteins. And for this, we use confocal microscopy for better quality images and to track intracellular localization of fluorescent signals that are associated with our ribonucleoproteins. So here we stained our nuclei with DAPI to visualize our cells. And here the fluorescent labels are ADO, and these fluorescent labels are attached to the tracker RNA on the RNP complex. As you guys can see here, The no treatment and free RNP groups show no fluorescent signals. However, we demonstrated cellular internalization of ribonucleoproteins by using lipofectamine. Here are the other three delivery systems that we use. We use hybrid peptides, cationic polymers, and planase. And these also showed varying degrees of efficiency in terms of delivering our ribonucleoproteins to our HEC293 cells, with hybrid peptides being the most effective and planase being the least effective. So here are T7 endonuclease 1 results. This is our DNA, or our CRISPR-treated DNA samples before T7 digestion. It shows a pretty strong band here, right under the 1,000 base pair. But interestingly, after T7 digestion, we see a much more faint band, which to us is interesting because it shows that our T7 enzyme is showing nuclease activity within our CRISPR-treated samples. However, we should see 2 bands within this region, but we're only seeing a much more faint band. And we're thinking that the DNA strands are being cleaved very small, and that's why they're running smaller on our gel. So some future directions I can take with this project: I can confirm gene silencing by confirming mismatched DNA by using our T7 enzyme digestion, and by showing that the cell is not functional through other assays. I can also study transfected cell phenotypical characteristics and show that protein signaling pathways have been affected. And lastly, I can try in vivo CRISPR-Cas9 delivery. So I would just like to thank Dr. Montazeri because he took me under his wing when I was looking for a cancer-related capstones project, and he helped me a lot throughout my research. I would also like to thank Riley Hall and Parvin Madapur because they were very patient with me when I was learning the processes, and I messed up a lot, but they were very helpful and encouraging whenever I messed up. So yeah, thank you guys, and let me know if you have any questions.
Great. Let's give Desmond a round of applause. Thank you so much for a fantastic presentation. Yeah, but let's open up for questions. We have quite a bit of time, so we can have maybe a few questions for Desmond.
Desmond, can I ask a question? Very intriguing data you showed there. These peptides that you are using, those were peptides, right? The delivery peptides, is that right? Have they, because you mentioned in the end of your presentation, you mentioned in vivo studies, have they been tested in vivo?
They have not yet.
What kind of problems do you anticipate when you move to in vivo with something like this?
Ooh, specifically for RNPs, because the protein is from bacterial origin, I would expect that there are some immunological responses that we see. And it might be pretty difficult to deliver these systems in terms of like delivering in vivo for RNPs.
Is there any way to circumvent that? That's my question, but I'm just asking myself, I guess. Very, very nice. Thank you.
Desmond, I just had maybe a question, and maybe I missed this, but is there a reason why the hybrid peptide is the most efficient or effective at delivering?
These peptides were specifically designed for our project. So I assume that's why it was the most effective in terms of delivering our ribonucleoproteins to our HEC293 cells.
I see, but then the polymers were just more generic.
Yeah, the cationic polymers and lipofectamine that we see here are commercially made transfection agents.
Oh, I got you. And that's probably why that's patented, right? There's some, there's some design associated with that delivery. Okay, thank you.
Can I ask a more general question, Desmond? So after you've done this and you've looked at how well this, I mean, CRISPR-Cas9 is not the only way to do this. After you've been through this, do do you still think CRISPR-Cas9 is the strategy to pursue, or do you think it might be better to go a different route?
Like, to my knowledge, I know of like talons and zinc fingers as like
Yeah.
DNA editing methods, but I think CRISPR allows for a more precise way, especially if we're looking at the route of gene editing in terms of repairing genes, because I've read clinical trials where they have successfully repaired gene function within, I think, Fanconi anemia patients. And I think this in the future could be a potential way of gene editing.
Yeah, I think you're right. I think the other methods, they can compete for disruption, but not for gene repair. I think you're absolutely right with that. Okay, thanks. That was really interesting.
More questions for Desmond? Well, Des, I'm going to ask you my question that I will ask everybody. I'm very curious, especially with your combination of psychology and the biochem. I'm wondering where you're thinking of going with your career moving forward after Chapman.
So throughout my undergraduate career, I was just purely focused on trying to get into med school. But after doing cancer-related research, especially this semester and during interterm, I think research is also a viable career option for me, and I'm looking into that. But overall, I would like to take a gap year and then apply to med school.
Wonderful, wonderful. I think certainly that the gap year will probably be very helpful to sort of like delineate what pathways make sense for you. But that's great. Well, let's go ahead and thank Desmond again for a wonderful presentation. We have about 2 more minutes, and I want to just make sure that we are good on time. So you can take maybe a minute break, and then we'll get to Peter as a last presentation for the for our session. And Peter, I just want to make sure when you are presenting, would you mind putting on the chat, just turning that on, because I will send you a message once it's like 10 minutes or so.
A chat?
Yeah, the chat box for Zoom.
Oh, yes. Okay.
Open when you're presenting.
Okay.
And just giving you were in here earlier, I just want to make sure that your slides can successfully share. So do you mind just sharing the slides and just making sure that
should I do that right now?
Yeah, that sounds great. As we're preparing.
Sorry, I am so technologically inept.
That's fine. I'm glad we have like some minutes in between, so we're good.
Ooh. All right. Yeah. Does that work? Yeah, that works. Can you see that?
Not yet. If you click on the green share screen button on Zoom, there should be a way to look for the particular window that you want to share. Oh, there we go.
Does that work? Okay.
If you try playing the slideshow, let's just make sure that it.
I just want to make sure. Is it showing the black screen or showing the big screen? I just wanted to.
It's showing the presenter view.
Oh,
so if you go to display set,
okay.
Actually, if you go ahead and play it again.
Play it again.
Yeah. And then if you go to display settings, I believe. Yes.
Oh, okay.
There we go. It's good. That's fine. I'm a computational chemist. I have a little bit of knowledge about how to deal with computers. Okay. All right. Well, that's actually good. I'm glad that we had that extra time. Peter, you ready to give the talk?
Yes.
Wonderful. Well, it is 12:39. We're in our last talk of the capstone presentation. I want to thank you all for your patience and also sticking around for all the talks. And so we have Peter, who is a biochemistry and molecular biology major. And Peter worked with Dr. Chang and investigated a study in hepatocyte regeneration, specifically looking at the major signaling pathways and potential therapeutic applications. I'm very interested to learn about this. So Peter, go ahead and take it away.
Thank you, Dr. Ogba. Good evening, everyone. I'm sorry for missing a lot of your presentations. So my topic is on hepatocyte regeneration and on the major signaling pathways and potential therapeutic applications. And just on the get-go, so hepatocyte regeneration after 2/3 hepatotectomy, which is actually the resection of the liver of 2/3 of the liver, and that's why it's called a partial hepatotectomy, is a novel mechanism for reestablishing liver mass and liver function. Although liver regeneration is widely studied, the major signaling pathways have not been clearly defined yet, as clearly. Through analysis of the signaling pathways, the mechanism of the regeneration can be broken down to better understand the possible therapeutic treatments that facilitate liver regeneration. And one of the things that I was able to find was the mesenchymal stem cell research, and it currently had a lot of avenues of actual therapeutic outputs, such as post-therapeutic treatments that you can use for liver transplantation. So it actually will help with the transplant rejection rates and actually improve that in some cases. And there's actual good clinical trials currently running on that. So it's very interesting on that end. And okay. And so, so I start, oh, sorry. Sorry, okay, all right. So yeah, so the immortal organ. So sometimes the liver at one point was considered the immortal organ, and some, like there's in the myths of Prometheus, where we explain things such as the tail of the eagle that would come and knock out Prometheus's liver, and then it will regenerate over in the next day, and it will be a constant cycle. This idea kind of is very interesting to me because they were actually talking, even in the ancient Greeks, they actually had this idea that the liver was somehow like regenerative or something that will just constantly come back and over. And so it seemed to have some idea even back then. And so it was very interesting to show in my case. And actually we see a lot of this in real life applications now when we see in living donor donations of the liver. And a lot of this, we see living donor donation. It's a process of donation in which recipient receives the organ and the organ resection, and the donor actually survives the procedure. So there's two people, one person donates, another person recipient. They both come out really good outcomes. And so, so that was the basis of my actual study based on that. And I thought it was very interesting to show what that was actually gonna, what aspects we can do to help that be a more success in the long term. And so when we see that, we actually see that after resection, the donor has around full functionality of the liver achieved in about 1 month in functionality, and actual full liver mass in even less than 6 months for some people. And so this is due to those multiple redundant pathways that are actually built into the liver pathways that actually facilitate cell proliferation upon injury. And we see that without injury, that the liver is actually pretty quiescent in the cell proliferation activity. But upon specific injury, we see all of these different redundant pathways that get activated to maintain regeneration to a certain extent, without the loss of functionality during the process as well. And so regarding that, in order to see that, we have to actually look at the understanding of what liver regeneration means. And in our case, actual regeneration is, to understand, is actual cell proliferation. And so, and cell proliferation is divided into two things, and into hyperplasia and hypertrophy. And to describe those two, hyperplasia is the actual increase in the amount, the increase in the actual size of the, hyperplasia is the actual increase in the number of cells that are actually, that actually gets produced. And what happens in hypertrophy is when our actual cell size, and you can see here where you see the actual nucleus is very small, and you see a lot of extracellular matrix. And so it's actually the actual enlargement of the cells itself, and that's what hypertrophy is. And so we got to understand, for our sense, this is actually not considered a true regeneration in the sense such as like the liver, I mean, not the liver, like a lizard loses a limb and some kind like that, and like that limb is actually being regrowth through an embryonic stage and bringing about an actual full functioning limb. But in this case, it's more the case that after you resect the liver, the remaining portion of the liver starts to go through this process that is actually more considered a compensatory hyperplasia. And it's where after, if you see this picture right here, we see that after lesion, we have a small portion, and that portion is able to maintain from what a portion has to maintain its actual functionality. At the same time, it is proliferating and actually make up the actual liver mass eventually, so that you will have a fully functioning liver for the individual. And so very interesting. And so, yeah, and so we go into looking into one of the first signaling pathways I wanted to look into is actually the protein tyrosine phosphatase liver regeneration, PRL1. And this we see here in the protein tyrosine phosphatase liver regeneration 1 is an intermediate early gene that is significantly induced during liver regeneration. Liver size and metabolism function and PRL mutations were comparable to controls, indicating that PRL1 is dispensable for liver development and postnatal growth. So the actual doesn't seem to do anything with the actual development part of it, but then we will see that actually what it does, it has a lot of instances of use in where it has proper timing of liver regeneration after partial apoptectomy. So after the resection of the liver, there is portions where certain cell cycle needs to be induced in order for us to go into the next cycle from the S to the G1 cycle, and G1 to the S cycle. And so this is really important to see because, oh, sorry. Oh, sorry. So yeah, so why is this important to know the timing of the liver regeneration after partial apoptectomy? And when we see the regeneration of the liver, the whole point of regeneration is there is a stopping point at a certain point, and there has different instances where it should be able to stop. And so how do we know when this foreign liver is in another person's body that it can actually be able to stop at a certain point? And there's actually these PRL1 is actually a good indicator of these types of I'm not quite sure. Oh yeah, these kind of compounds, what it actually does. And so it might actually help you to understand that there's a stopping point, and it actually helps to actually do the proper timing and also the actual proper termination point of when of the proliferation process at that point. And so it's good to know those two in order to actually understand when it will stop. And it actually is very interesting to see that in a lot of cases, depending on who it is, the actual liver will regenerate to the size of the actual individual that it has been received the actual organ. And so this process is actually pertaining to that instances. And so, okay, and so we'll look at the first signaling pathway. After that is the FGF19, which is fibroblast growth factor 19. And we see that in IL-1 beta inhibits the beta klotho expression of FGF19. And in saying that, we say the FGF19 subfamily acquires the klotho protein as a co-receptor to facilitate their interaction with the FGFRs. And here we can see a lot of, I found a good example of this reaction, and they show the FGF19 going, meeting that KLB, which is the beta klotho expression right there. And so that really needs to be the co-receptor in order for it to be phosphorylated in this next step here, and requires the FGFRs in system to actually help phosphorylate in the end the actual ARK1/2, which is the extracellular signal regulated kinase. And that will actually phosphorylate that, and it will, if you see the system here, this is showing a system where FGF19 is actually proceeding through. But when it is phosphorylated, it will actually, instead of decreasing the apoptosis, it will increase it. And so then you will have this attenuated system in liver regeneration. So from all this, from what we got from this is that these results suggested that the inflammation and IL-1 beta was actually playing an important role in regulating that and regulating FGF19 signaling and function in the liver. Okay, and so we go on to the next one, which is SOCS1/3, which is suppressive cytokine signaling 1/3. And we see for this one that IL-2 mediated liver actually upregulates the SOCS3 into overexpression, leading to an attenuated liver regeneration. And we see what happens is the IL-2 increase the mRNA expression of the suppressor, and the pro-inflammatory cytokines such as the IL-6, and here in this case the IL-22, are all actually are causing the overexpression, is actually causing to actually take away the actual IL-2 induced STAT activation from it, and actually decreases the whole IL-2 mediated liver cell regeneration process. And then we have actually the miRNA 155 overexpression, which downregulates the SOCS1, promoting the hepatocyte proliferation. And we see in this actual article, they were showing that there was actual knockout of this mRNA gene, which actually caused the actual therapeutic application of mRNA was actually observed. And so we had we saw that actually knocking out this gene had a pro-proliferative effect on the regenerative hepatocytes, targeting that suppressor cytokine. And so it helped actually, so it actually helped more of the pro-proliferative effects on the regenerative. Yeah, I just repeat myself. But yes, and so the next one we have is the estradiol. Is another another compound is widely recognized as a stress signaling, and pH has been has shown to induce estradiol production as an initial response to the resection prior to the proliferation molecule. So this estradiol we see that it happens when there's actual an injury or some kind of response to injury is produced in the liver. I'm not sure if it's specific to the liver, but it does produce in the liver, and so it had a lot of post-therapeutics as well, where it can actually help accelerate liver regeneration through the estrogen receptor alpha. And this showed a lot of possibilities for another additional post-treatment for transplantation, after transplantation. And oh God, I knew this was going to happen. I was trying to lower it. But and the final thing I really wanted to get into, the mesenchymal stem cell therapies, is really the actual gung-ho part of like a lot of the things that's going into the clinical trials right now. It's very interesting to see. And there's like, first of all, mesenchymal stem cell cells originally was like the subpopulation of multipotent and non-hematopoietic cells initially isolated from bone marrow and bone marrow. But now I think it's done a little differently. But yeah, I'm not quite sure if it's from bone marrow anymore. But and so yeah, we had a lot of this was very interesting to see, because they were talking a lot about the healthy tissue proliferation and understanding, like, a lot of the ideas of one of the research they actually wanted to see if it actually have helped the cell regeneration. And there was they saw certain issues that when they actually did that, there was actually conflicting issues with fibrosis. And there's this whole actual problem to understand is that cell proliferation to actually make up the mass compared to the actual fibrosis, which will actually attenuate the actual function of the liver. And so it's almost kind of like thinking of something like scar tissue. And so having too much scar tissue in that region, it will actually cause fibrosis type of disease with the liver. And so that was a good thing. You got to have a certain tissue homeostasis in order to balance that, in order to understand that. So from just based on what they were describing there, it seemed like it was not a great aspect for treatment for cell proliferation, but there was other studies that actually went into the other kind of, there's immunoregulatory aspects of this actual this research of the stem cells. And so we found that eventually a lot of the clinical trials that they went into, they actually went into a lot of the immunomodulatory cell therapies, and which was in a way a compensating together where you do an immunomodulatory cell therapy with the complementary plan where you have the pharmaceutical immunosuppression along with the immunomodulatory aspects of mesenchymal. It might be a good thing to like do a kind of a combo where you can actually be an additional thing that you can add on. And so it will help. I feel like if we, a lot of the clinical trials I was going into, it seems like they are finding ways that can actually bring this into more mainstream medical field. So that was very interesting to see. And yes, sorry, I will end it there. So these are my references.
Great, wonderful. Let's give Peter a round of applause. Thank you so much. Right on time, by the way, Peter. I didn't mean to like stress you out. Like,
Peter. I'm so sorry.
Oh no, don't apologize. You were like, it was right on time. Yeah.
Yeah, I was lowering it and lowering it. I was like, I by the time when I was first doing it, it was just like so long, and I was like, oh my god, how do I lower this? And but yeah, even then, so a lot of topics. Yeah.
Yeah, giving time presentations, it's a skill that you have to.
Yeah, I'm learning, and it's so nerve-wracking. And
no problem. Yeah, you did a great job. Okay, let's open up for questions. We have a couple of minutes, and so go for it.
Peter, that was wonderful. Thank you so much. I appreciate that. Very, very interesting and important research. You mentioned hyperplasia and hypertrophy. I'm perpetually confused about those, and I should know better because I'm in cancer.
I am so glad you said that because I'm constantly confused about that too. But it takes me a while, but
people seem to be using those terms all over the map.
Yeah, like just interchangeably. I think I've used it interchangeably too, and so. But then from this actually looking at it, because they go into specifics on cell proliferation, they want you to differentiate between those two. And it seems to be a very important aspect of the liver that it does hyperplasia compared to hypertrophy. Where
and my question, if I can ask that question, yes. There was nothing about dysplasia, right? You never in your
no
dysplasia.
Not dysplasia. I did not write that. Yeah, not dysplasia.
Because that would be leading to a change in the tissue structure, which is not supposed to happen during regeneration, right?
Yeah. So I feel like what happens with there is actually a actual certain type of liver disease that actually causes to do this direct hypertrophy, where it just starts to swell up, and that's not quite sure which disease. I didn't focus on all the liver disease, but one of the liver disease has that kind of symptomatic behavior. And so it seems like it's not a good thing, is what they're saying. Like you need to actually have like a percutaneous catheter in to actually displace that fluid. And like it seems to be something that's not something the liver should be doing.
Okay.
And so they were explaining how liver regeneration is quite not technically the right term to call liver regeneration, because it's not regeneration in any aspect of it. It's the actual technical term that they would use is the compensatory hyperplasia to be the correct term for what that facilitation of the liver to be full.
Thank you.
Size. Yeah.
Thanks for clarifying. I do have another question, but somebody else should go.
Is that maybe Dr. Bisalfi, you can probably continue.
So these signaling pathways that you presented, were they found? How were they found? This is in vivo in patients, or was this in vitro studies based? You know,
it was. I believe it was in. I looked. It's through mouse models. So in vivo would probably be correct, right? Sorry. Yeah. Yeah, so it was through mouse models, most of to see where, and it was a lot of tracking. Some of them had to do with more like the what is the word, the radioactive tag type of situation, fluorescent dye situation, where they did fluorescent markers to show certain things in population. But this one was.
Yeah, that makes sense that it was discovered in mouse models. So yeah, thank you for that clarification. Well done once again.
Thank you.
Wonderful. We are at 12:59, and I'm just proud of myself. I'm sorry, I should not praise myself for a great moderated session. Let's go ahead and thank Peter for a wonderful talk, and let's actually thank all of our speakers for the entire day. But I want to highlight Dylan, Brian, Hannah, and Desmond. So let's go ahead and thank all our speakers for a wonderful presentation.